all to the second day of the symposium.
Hello,
and hi, Amitabh, how are you? Yeah, all good. Can you hear us? Are you audible, Amitabh? Yeah, that's okay, I guess. So, good morning, Alex. Good to see you, and good morning, everyone. Good afternoon, good evening.
Morning as well.
I'll just move out of the way, and before I do that, I'll ask Rahul Pal, my senior colleague who works on autoimmunity, to be the moderator, coordinator of the day. And we look forward to this exciting second day. It'll be as exciting, hopefully, if not more, as yesterday. And we got a very good feedback yesterday from students and faculty alike. Thanks very much, and have a good time.
Hello, my name is Rahul Pal, and I'll be moderator today for today's session. So our first speaker is Dr. Alex Sherek. And Dr. Sherek is a core member of the Institute for Medical Engineering and Science and associate professor of chemistry and an extramural member of the Koch Institute for Integrative Cancer Research at MIT. He's also an institute member of the Broad Institute and a member of the Ragon Institute, an assistant in immunology at Massachusetts General Hospital, and an instructor of the health sciences technology at Harvard Medical School. Dr. Sherek received his PhD from Harvard University in chemical physics. His lab research is directed towards development and application of new approaches to elucidate cellular and molecular features that inform tissue level function and dysfunction across a spectrum of human health and disease. And some of his work, his lab has identified ACE2 and transmembrane serine protease 2 co-expressing cells within lung type 2 pneumocytes, ileal absorptive enterocytes, and nasal goblet secretory cells. And he's also found that ACE2 is a gamma interferon stimulated gene, the implications of which he might talk about today, or he might touch upon today. Dr. Sherek, please.
Good morning, everybody. Thank you so much for having me. Sorry I can't be there in person, but I guess it's appropriate that we're having a seminar about COVID when we can't be in person. So let me share my screen and make sure you can see my slides. Everything good on your end? Tell me when it comes in. And are you seeing the right version of my slides right now?
Oh, you're not on presenter view yet, Alex. Oh, you are. You're fine now. You're good.
Hold on. Did I swap it to the wrong display, Shiv?
No. You were okay. I'm sorry. You were okay a minute ago. Yeah, great. Thanks.
Yeah, I think this is fine.
This looks good.
Perfect. Shiv always trying to mess me up. Anyway, thank you so much for having me. It's a pleasure to be here today virtually and tell you a little bit about some of the work that our lab, like many others, has unexpectedly been doing over the past couple of years to help gain insight into COVID-19. I think that we can all agree as scientists that when something like the COVID-19 pandemic arises, each of us feels a moral obligation to do whatever is within our power to try and help gain insights into the disease. And I'll tell you about the things that we've done in our lab, and I'd love to hear more about some of the things that you all have been doing, but I can just relate, personally, many of the people that I work with felt very out of sorts and as though they were, unable to do anything that would be beneficial. So they felt powerless. And over the past several years, we've tried to do the things that we could to gain additional insights to help make strides forward in our understanding of COVID-19. So I'll just start off by saying, COVID is a global pandemic. I don't think that I need to dwell on this, hundreds of millions of cases, millions of deaths, and, impacting basically every sector of the world at this point. If we think about what we sort of knew at the start of the pandemic, what we really understood were sort of the clinical presentations of COVID-19. So we understood that it led, usually to upper respiratory tract involvement, cough, and the like, that there were gastrointestinal symptoms in a set of people, but there was this general inflammation and malaise that showed up across a number of individuals. Some incredible work that was done building off what had previously been studied in SARS-CoV-1 quickly identified that ACE2 was the receptor for the virus, and that it used, in the main, the accessory protease TMPRSS2 as a way of entering into cells so that it could begin the viral replication cycle. And so, whereas we could figure out very quickly how the virus was, entering cells molecularly, we could understand at the systems level how people were feeling. the thing that we began to wonder is if we could start to help connect these two levels, these sort of molecular and systems level behaviors, by using some of the technologies and data that we had on hand. And so you might ask, why did we think that we might have the opportunity to help here? Well, over the past several years, our lab, along with a number of others, has been involved in doing single-cell genomic characterization of a number of different tissues. And I'll explain what that means in a second. But in short, what we had done is we had made these beautiful cellular maps of multiple different tissues across multiple different species, really going in and looking at each individual organ and then deeply characterizing the cells that comprise it, and not just looking at one property of those cells, looking at the expression of all 20,000 genes. So just in a nutshell, what we've been doing that we thought could help was we've been taking a number of different tissues that were impacted by different diseases, isolating cells from them, and then taking advantage of advances in micro and nanotechnology to isolate individual cells and things like reverse emulsion droplets or small picowells, so that we could then, within these, capture the messenger RNAs of every single cell using some very simple oligo DT priming, and then amplify this up and create a sequencing library. And what this does is, just like in bulk genomics, it lets us go in and for each of these individual cells and each of these tissues, characterize the expression through sequencing of basically all 30,000 genes within each element. And so if you take, for example, 100,000 cells, you end up with 3 billion data points that you could use as a way of trying to say which of these cells might be relevant to SARS-CoV-2 infection. And so, from this big data to getting an idea of what's happening, this is sort of the steps. It's a little bit computational, but for those people that are interested in the audience, we basically take this big data matrix, filter through it, identify genes that are variable, and use a lot of this to find cell types. And then, by basically creating a social network of cells, pulling together cells that are similar, pushing apart ones that are different in gene expression space, we get to these maps that help us to understand the types of cells that might exist in the body, like the one that I showed you at the beginning, this sort of beautiful hairball. And to help with interpretability, what you can think now is that each of these points in this graph is a single cell. This right here, these connections are sort of the relationships among them. And so for each individual population, what I have is sort of this vector of expression of, 30,000 genes, where I can say gene 3 was expressed, but gene 1 wasn't. And from those genes, I can identify what the cell types are. So I can find genes that will help me to figure out that I'm looking at microglia or eosinophils and the like. Some only exist, for example, in the brain, and some, the immune cells shift broadly across multiple different tissues. So, you might say, well, why did we think this would be useful? Well, one of the genes that exists within our matrix is ACE2. One of the other genes that exists within our matrix is TMPRSS2. So what that means is we can now start to say, well, where in the body are we seeing expression of, this entry receptor in the accessory protease? And so we can see, for example, that there's high levels of expression in the ileum. So in the jejunum, matching what we'd see at a clinical level, we're also seeing it in the lung, but not in some of the other tissues. And we can dive down a little bit more, looking at individual tissues, like, for example, here at the lung, where now I've done a different type of plotting. Each point is once again a cell. I group cells by similarity, and you can see that, we're recovering fibroblasts, type 2 pneumocytes, macrophages, so on and so forth. And within these, I can say, well, which are the cells that are positive for ACE2, and which are the ones that are positive for TMPRSS2? And what we see is that we're getting detection of both in a subset of ciliated cells and type 2 pneumocytes. I think that what's interesting about this, when I was first looking at this data, that made us think that this couldn't be right, is that, we were always finding cells that co-express. So in the nasal epithelium, we find secretory goblet cells or ciliated epithelial cells. In the lung, type 2 pneumocytes, or in the guts, adsorptive enterocytes. But the numbers of cells that were ACE2 positive or ACE2 TMPRSS2 positive were really small. And this didn't really make sense in the context of sort of the severity of infection that we were seeing. And so, what we're really seeing were needles in a haystack. We began to wonder, is this really what happens? Is this data really useful? What are the real targets of infection? What are the consequences of it? So, in parallel to some of the work we were doing, there was some beautiful work done by others, really taking tissues ex vivo and infecting it and trying to find cellular targets, or looking in vivo at which cells were positive for virus. And, the difference between some of these methods and some of the stuff that I'm showing you now is that these really, were focused on particular molecular markers because of the difficulties of doing broad unbiased profiling in vivo or ex vivo. And so it gave us an idea of what might be relevant, but it didn't really help us figure out which cells were truly infected and what was associated with it. And in parallel, there were these beautiful studies showing us that in the periphery, that elevated inflammation associated with severe disease. Oops, sorry, I hit the wrong button, wrong direction. And that type 2 or type 1 interferon responses have been shown to be important in the periphery. But there were still open questions about whether or not these models were correct, and really what was happening in the target tissues of infection. And so, what we wondered is if we could help to get into this using some of the same technologies that I had already introduced. And so many of you probably have, unfortunately, at this point, had many nasopharyngeal swabs or NP swabs, which is commonly the way in which you go about looking for virus. And so many of you are probably familiar with the idea of having a nasal swab, using it to collect virus, and then putting it into a tube to see whether or not you're positive or negative by qPCR for it. Well, the thing about these swabs is that at the same time that you're swabbing for virus, you actually end up sloughing off some of the surface of your nose right here. And if you think about it, this is really the first site with which the virus makes contact with your body and can drive infection. And so what we began to wonder is if we could take the cells that existed on this, came along for the ride with the virus, do the same kinds of technologies that I had showed you previously, sequence those, and use this as a way of characterizing sort of the initial targets of infection and sort of the differences between disease severity. So to make a long story short, we developed a protocol, as crazy as it may seem, to isolate individual cells from these swabs. And so I'll refer you to it on protocols.io, but just note at the end of the day that we figured out how to actually get out a variety of different cells, whether they were ciliated cells that I'm showing you right here that could swim around on our cover slip, to squamous cells, to goblet cells. And we collected swabs from a good swath of individuals ranging across a variety of different disease states, both those that were negative, so some healthy donors, as well as some non-COVID ICU incubated controls, and then individuals that were SARS-CoV-2 positive, whether mild or moderate or severe or critical disease. And I'm defining that based upon the WHO score over here. Our controls, were people who needed intubation but did not have COVID-19. So we got a good representation of the local demographics in the place where we were partnering, which was with Sarah Glover's lab down at the University of Mississippi Medical Center. Good blend of genders, good blend of ages, and ethnicities. And from this set of about 60 swabs, we were able to recover about 33,000 cells, which I'm showing you over here, much like I showed you previously, where what you can see right now are each individual point is a cell. They've been clustered once again by their gene expression in this high dimensional space. And what I want you to see here is that we're getting a good representation of the multiple different cell types that you might expect. So just to orient you really quickly, we had these basal cells, which are the stem-like cells of this tissue, going down through some of the other epithelial cells that play a role in the nasal barrier, including goblet cells, these squamous cells over here, and these ciliated cells that actually help to sweep things out. We also are capturing some immune cells, some T cells, some dendritic cells, and for the aficionados in the group, cells like ionocytes and enteroendocrine cells that have been more recently described. Now, this is sort of the map of what the nose looks like across all these individuals. What you can now do is you can take this and you can overlay whether or not the individual was positive by PCR for SARS-CoV-2. And what you can see is that there's some places where things seem to broadly overlap, like up here and here, but there are places where there's a real separation that we're seeing between disease and absence of disease. And really, that's less on the immune cells down here and much more on these epithelial cells that I'm showing you up top. So what we decided to do was to take away the epithelial cells and redo some of our analysis, focusing specifically on them. So this is a similar map, slightly different because it looks just at genes that are important for epithelial cells. But once again, what you can see right here are some of the epithelial populations that are important. Basal cells down at the bottom and some of these more mature differentiated cells up top. To really overlay the differentiation trajectory on top of this data, what we can do is we can take advantage of some of the properties of the RNA that we have looking at splicing. So this mechanism called RNA velocity that basically looks at the ratio of unspliced to spliced RNA to figure out whether or not a transcript is new or old. And that gives you an idea of what the cell is trying to produce versus previously produced. And knowing what a cell wants to do can help you to describe, sort of a direction, a flow across this data set. And so what this suggests in our instance is that Just like you might expect from the textbook, that if we start down here at these basal cells, that we're going to differentiate up into some of these more mature populations like goblet cells over here, going up all the way through to some of these developing ciliated cells, and our squamous cells over here seem to sit separately, as do our ionocytes. But we do recapitulate, sort of what you would have imagined out of, some of the papers describing development within this tissue. So, if you're sitting here, you might ask yourself something like, what changes under SARS-CoV-2 infection? Well, if we start to look at this map, what we find is that there are some very significant changes. One of the biggest changes is that we get this huge increase in the number of secretory cells that we have. And when we look at them a little bit more, we find that they're even a little bit more divided in their function. And so you see that there's a significant increase in the fraction of these cells that show up within this tissue, probably suggesting an attempt at clearance of virus from the tissue. And this is something that we can go back and validate in some independent controls. So if we sort out some, epithelial cells from some additional swabs and stain for markers of, of secretory lineage that I'm showing you right here, CCAM6, we can see that there's a significant increase in the tissue among the CD45 negative cells. What you might say, well, if you're getting more of these cells, there probably has to be a loss of something. Well, there is. What we find is that actually we get a loss of these mature ciliated cells. So you get sort of this decrease in these cells that would normally help to move things out of the nose. And then there's some other things where you see actually an increase in some of these precursor cells, which sort of, suggested to us that actually there was maybe this de-differentiation process that's happening where we were losing some of the most mature cells under these inflammatory conditions, getting more of these intermediates, and then getting broad differentiation of these populations right here. Now, this is really what's happening sort of at global level. You might say, well, what happens in viral infection if you focus within a specific population? What changes within the ciliated cells? Is it just abundances, or is there phenotypic differences as well? What we find when we go look is that actually, there's something surprising. If we look within ciliated cells, for example, comparing, our controls to our mild disease or to our severe disease, and look at the genes that are significantly upregulated in infection, we see a bunch of probably many of your favorites, IFITs, MX2, all the things that you would normally associate with canonical interferon responses. We see exactly the same thing if we look at severe disease over here. But you notice that some of your favorite genes aren't quite as high. And when you go and you compare what happens in severe disease to mild disease, actually there's this really interesting difference where it looks like there's a lack of strong interferon response that emerges within these individuals. So there's a significantly higher expression of some of these interferon genes in the context of mild disease relative to severe disease. And this wasn't just something that showed up in these ciliated cells. It actually turned out when we looked across every single one of these populations that, there was a much higher intrinsic interferon response in the epithelia in individuals that had mild disease. So across goblet cells, ciliated cells, secretory cells. So it looks like there's sort of this severe blunting of the antiviral and interferon response in more severe disease cases. And, we can see this if we go look at some of the major genes that would be associated with interferon response as well. Like, for example, looking here at STAT1, we can see that there's a significant increase in the activation of STAT1 among those with mild and moderate disease. And, what's interesting is that even if we look at individuals that came in with the same scoring, so that had, similar strength at swab, we can find that among these mild and severe that, interferon was depressed even when they were still at this early stage. So right here, I'm showing you points that make this up. And what I'm telling you is that if somebody came in and tested when they were mild, but went on to have severe disease, that we could already at that point tell that, they had a decreased or a decreased antiviral response, suggesting that this may actually be a biomarker or a mechanism of infection. So you might ask yourself then, who is producing interferon? Where is this coming from? And so as we look through our data, we actually don't detect a lot of interferon. We get a little bit of gamma, for example, in some of our T cells. We get expression of many of the inflammatory cytokines that you might think about, but not a lot of interferon. It's really pretty sparse on this side. Now, you might say, well, is that just an issue with detection in your technology? Maybe, but when we go look at comparable samples from influenza infection, for example, we can get pretty good detection of interferon alpha and beta, suggesting the technology we're using is really capable of picking up, these genes like interferon alpha and lambda, but we're really not detecting it in COVID. And, this is consistent with what others are seeing in the field where, it really looks like there's a much weaker interferon response to SARS-CoV-2 relative to, other infections like influenza. So, in human bronchial epithelial cells, you see that SARS-CoV-2 leads to a very small induction of interferon, if any at all. And once again, in the plasma of individuals that have COVID or influenza, very low levels of peripheral interferon lambda. And, this also lines up with what some individuals were seeing in the periphery. Out of, like, for example, Max Krummel's lab, where they were detecting differences in sort of the activation of ISGs in the periphery in individuals that have mild disease versus severe. And what we're saying is that we're seeing exactly the same thing in this initial target of infection and seeing it early, suggesting that it might be important. Now, you might ask what leads to reduced interferon. Could it be something like autoantibodies? You probably have read that about 10% of people with severe COVID have autoantibodies against infection. Well, in our data, we get that 1 in 10 that I'm showing you right here at the top, but it doesn't associate with severity at all. You might ask if it's something else, some of these, inborn errors of immunity that, that John Laurent and others have talked about from the Casanova lab, doesn't seem to explain what we're seeing in our specific populations. The other thing that we began to wonder as we started looking at things is, could we now go refine the viral targets that we have? So in the first part of this, I told you that we were using existing data to figure out what would be likely targets. And so far, I've told you the impact of infection, but I haven't really told you which cells are infected and what are bystanders. Well, one of the nice things about the technologies that we use is that they capture polyadenylated mRNA indiscriminately. And so when we take these swabs, we can not only get eukaryotic mRNA, but we can also get at that of the virus as well. So if we go and we take these tubes and we take our cells and we align to virus instead of just human genome, what we can do is we can find viral RNAs in individuals that have mild and moderate disease and severe disease. And nicely, we don't detect any virus in healthy or non-COVID ICU patients. And so here I'm really showing you the number of viral reads we're getting for each individual cell. And overall, we're getting about 3% of cells are positive. You might ask, is this quantitative? Well, with Party Svetti's lab, we did some work, quantifying how this compared to sort of, standard virologic measures like RT-qPCR. And you can see we get this beautiful linear correlation showing us that we're quantitative in our detection. There is some stuff that we have to do in our sequencing to correct for ambient RNA contamination. You can imagine we take its tissue, we break it apart. There's free-floating virus, and so we spent some time correcting for, what might just be ambient RNA or ambient virus relative to what was within cells. Beyond that, we can actually even take advantage of the viral life cycle to go a little further. You probably all know that this is a positive strand virus that is it's a single positive strand of RNA. It then goes through this viral replication to make a negative sense strand, and then it goes through the splicing to create these subgenomic nested transcripts, which create these unique fragments that we can then align our reads to. And from this, what we find when we go and look at stuff, just to make a long story short, given time, is that really the cells that are positive are aligning with the ones we thought. There's some ciliated cells, including a weird population of ISG high ciliated cells, goblet cells, and helix cells, and these are really the cells where we have high viral coverage, good coverage of the viral genome, so many of the splicing variants, detection of negative strands suggesting that actually it's actively replicating, including some of the spliced variants. So how does this correspond to what we predicted? Well, actually pretty well. If we look at the fraction of ACE2 positive cells that, that show up in what we had originally predicted in our 2020 paper versus what are viral positive by this data that shows in our 2021 paper, what we can see is that actually there's a nice linear correlation between the two. There are some cells that are off diagonal up here, like interferon responsive macrophages. But when you look at it, you don't see a lot of splice virus here. And what we really think is happening is that these are cells that are, phagocytosing dead cells. And so they just have virus within them. Beyond that, you might ask yourself, well, what's different about cells that have virus? If you go and you look at what's higher in cells that are viral positive versus negative, can you find something that associates with a viral level that might be targetable to help prevent infection? Well, if we go look, what we can see is that within our SARS-CoV-2 positive cells, there are a number of genes that are upregulated relative to those that show up in our bystanders across each of our populations. On this side, you see many of the antiviral response genes that you would normally have thought about if you look through this list, as well as the master transcription factors, a lot of antigen processing via MHC1, some cholesterol biosynthesis, which interestingly connects back to some papers that were recently published in the literature. And on the side, if you think about what's higher in bystanders, interferon receptors, so maybe this makes them more responsive to the limited amounts of interferon that are there. Some interesting antigen presentation via MHC2 that lines up with some recent work that people have shown in the context of Ebola. Now, you might say, well, is what you're talking about really just driven by the presence or absence of virus? Well, what we can do is we can go convince ourselves that the level of viral infection is not driving some of the significance that we see in our data. So if we go look at our epithelial cells, we can see that we're actually detecting a similar number of SARS-CoV-2 positive cells, both in mild, moderate, and severe disease. Similar amounts of positive strand and negative strand. And even when we look just at infected cells within a specific population, we see that once again, really, there's a strong upregulation of these antiviral, type 1 interferon responses within mild and moderate disease relative to severe disease. Whereas on the other hand, if you ask, what's up in inflammation, it's really, some of these inflammatory genes like S100A. So in a nutshell, what I hope I gave you really quickly over this was that, you can now take some of these technologies and look at these samples that you would have never imagined looking at before to characterize certain impact of disease. We generated this resource that was useful for understanding what's happening in COVID-19. It let us look at the epithelial trajectory. We found changes in the abundance of specific cells and in intrinsic interferon responses that associated with disease severity within SARS-CoV-2 positive individuals. In the last minute or two here, what I wanted to do is just ask, how does this relate to what happens in the lung? So this is up in the nose, but we know as you go to severe disease, things progress down into the respiratory tract. Well, to address this question, we work with a number of partners across the Boston area to do this Boston COVID autopsy study, looking at a number of different tissues and asking questions like what changes in disease relative to healthy tissue, what cell types are affected, many of what I showed you before. And so we can dive right into the lung, use many of the same approaches that I showed you in the past, identify cells, and there's some complications that happen in the molecular biology that I'm happy to talk through. What we find here, once again, is something that's consistent, which is sort of a loss of some of the mature epithelial cells here, AT1 and AT2 cells, and some increases in some pathological intermediates. So this dedifferentiation like phenotype like I showed you before. In the context of the lung, we see some epithelial cells infected mainly in severe disease. They're so depleted from death that where we really see a lot of virus is in the myeloid populations. And here, once again, it doesn't look like it's spliced. So it really looks like this is, the macrophages clearing things up. And I'm happy to talk more about this, but, given time, I'll just step away. And I'll say that, I think really at the core of what we do is this idea of thinking about human biology and then using models to try and accelerate interventions back into humans. And so the other thing we've done in parallel in the context of COVID-19 is to try and characterize what happens in, mild infection in non-human primates with Dan Baruch, looking, for example, longitudinally at lavages from the lung to see, what are the dynamic responses to infection as you get high virus and you get decline. Some interesting things emerge here that I'm happy to talk about. And then, thinking about other models like the Syrian golden hamster, which, unlike the monkey, experienced severe disease. And here, once again, we see something very similar to what we see in humans with a loss of some of these more mature epithelial populations and a rise in specific immune states. All the data that I showed you is publicly available. And there'll pretty soon be some new data that we've been generating in the nasal swab studies, looking not just at sort of the original strains, but now looking at Omicron and Delta viral strains, and also looking at the impact of infection on epithelial responses. And I'll just give you a preview and say there's some really weird and interesting things that happen within the epithelial cells in those strains relative to the original one and post-vaccination. So with that, I'll just thank the team. And really, I want to thank these individuals who are really the people that drove all of this. Carly, who was the tip of the spear, my partner in crime, Jose, who used to be a postdoc in the lab and, set up his lab during the pandemic with whom, did all this work collaboratively. Andrew, Vince, Josh, Michaela, and Riley, who, despite all of my pushes to focus on their own safety, decided they really wanted to get involved and, worked in the lab during the pandemic. I'll thank a small list of people that were involved in doing all these studies, because these are things that happen through collaboration all around the Boston area. Spent a lot of time trying to figure out how to get involved, but also be safe. And we'd also like to thank, the study participants and their family, without which this wouldn't have happened, and our funders. And I'll just say thank you for having me. And I'll pop out and I'm happy to answer any questions you might have about what we've done or where we're going.
Thank you, Alex. Thank you very much. That was wonderful. Questions? I think Dr. Salman can start us off.
Can I?
Come here. Yeah. Please hold on just 1 second.
It's all good. Take your time.
Hey, Alex, wonderful talk. Really thoroughly enjoyed it. Quick questions. These samples that you have acquired and analyzed, these are a sort of snapshot of time, and you wouldn't know the initial infecting virus titer while distributing across different cell lineage. And I'm wondering how one would approach to normalize for that uncertainty. The time you are getting the samples, the virus titer might be similar, but this may not reflect what was the initial viral load that one individual was exposed to.
So it's a great question. So actually, what we can do is we can use some things to try and time it, to try and figure out what is the rough window over which infection occurred. But you're right, there's sort of this uncertainty of a couple days. And we really wanted to set up this study where we were going to be longitudinally swabbing people in the hospital, getting nasal swabs, and then looking at first testing and then go back and look previously. But we couldn't do that here. I will tell you that when we tried to do things within windows to look roughly at the similar time point, there weren't major changes in the amount of virus that was within the noses of these individuals. We do in the autopsy cohort see major differences in the amount of RNA based on timing in individuals that unfortunately succumb very quickly to infection. We detected much more virus than in those individuals that passed away after, say, 4 weeks. So if you look, there's this exponential decay in the lung of the amount of virus that we see. We didn't see that in the nose, and I think it's just because we were looking relatively early. But you're right, there is sort of this uncertainty where it could have been, 7 days or 10 days post-infection, but there were no discernible patterns in the data we had. But the answer is that what we're really hoping to do next with some partners all around the globe is to set up some pathogen readiness work where we do something similar to what I showed you here. We swab the nose, maybe collect some peripheral blood and do it longitudinally, and then look at, when somebody tests positive for either SARS-CoV-2 or another infection like RSV, we can detect not only what is, infected, what's changing, but go back in time a little bit. Unfortunately, when we tried to set up a number of these studies, the cyclic nature of the virus got the better of us. So by the time we were all ready to do this at a hospital in Belgium with some partners, cases had dropped to the point where we could no longer get a significant number of people in the study. So it's something that we want to do. I can tell you that on the other hand, in the animal models, like when we look at the NHPs or the Syrian golden hamsters, it's pretty consistent. You always get one outlier monkey because there's always one bad monkey, but going back to the animal model data where you can be very controlled, you don't see it. The most interesting things that we see there really are things at later time points where, where you see strong divergences. And even when you have like prophylactic IgG, you see that, there are these strong inflammatory responses that show up that are unusual. So some cool stuff there, but it's really not in the early time point. It's really in what happens in sort of that second phase. And I think it has to do with sort of early resolution versus sort of ongoing replication that leads to sort of divergence and severe disease.
So one more quick question. I think you may have the data with you, but it's just some curiosity about the bystander cells and those that were infected. Any difference with respect to, let's say, cytokine expressions or even the gap junctions protein? You might have some answers already.
Sorry, I did this really quickly. So in one of my slides, I showed that when we looked at, infected cells, that there was really a strong upregulation of a number of like ISGs within those cells. And then in cells and, things like MHC1, whereas when we looked at, cells that were uninfected bystander, we saw higher interferon receptor, suggesting that those might be the cells that are more sensitive, more able to gobble up any interferon that's around. We also saw, upregulation of MHC2 machinery. And there's been some studies that have shown that you could like sequester virus in endocytic vesicles that might relate back to. So, actually you're not getting, productive infection. But it's one of the things that we looked at, and there are some things in that list that, we're hopeful we could target as a way of actually improving, protection against virus more broadly within the nose.
Yeah, thank you, Alex. Thank you very much. I'll hand it over to Rahul.
Okay.
Nimesh, you have a question? Nimesh, go ahead.
Yeah, thanks, Rahul. Thanks, Alex. It was a great talk. I was just wondering that did you get a chance to look at some of the asymptomatic individuals? Is there a difference in the spread of virus across the cell types, or is there any kind of unique enrichment of the immune responses in those areas?
So it's a great question. We had some individuals that that were asymptomatic within our mild cohort, the ones that are 1 to 5, that they just got caught because of random testing. And our numbers are small, so I can't tell you, if there was something significant, but to the number of samples that we had, which, albeit is not huge, there was no major difference among them. Just very good, strong interferon responses, suggestive that, sort of early protection in, the barrier in your nose might tell you a lot about whether or not you're going to progress to more severe disease. And you might, appreciate then some of our questions that we have now, thinking about with vaccination or previous infection when you get reinfected, are there changes in sort of those intrinsic epithelial responses that might play a role? To what degree do immune cells come in? So we've just run a large study to address some of these questions, which we're currently analyzing data on and hope to share soon.
Thanks.
Dr. Shashidhara, would you like to ask a question?
Yeah, I have a quick question. I mean, more of a cell biology question.
Yep.
When virus titer, considering that irrespective of variations, it will be still a large number of viruses. How much of the AC2 distribution on the apical surface and the AC2 polymorphism in different cell types has an influence on the virus entry?
So it's a great question. In what we see, we're doing everything in dissociated cells looking at RNA. So an important thing is to go back and think about what the protein distribution looks like within the tissue. We did do some immunohistochemistry to go back and characterize this, but more limited cases to prove that where we were seeing ACE2 was actually where ACE2 was in the tissue. We did not go and look at the distribution, but it's a great question. We didn't have enough individuals in our cohort to really ask about how some of the genetics might associate, other than to ask simple questions like, are there mutations in some of these intrinsic antiviral genes that might play a role in explaining why there was these interferonopathies. But it's a great thing to think about, and it's something that we'd like to look at in the future to try and connect this back to infectability, but on the other hand, also really understand why there are differences in regional severity. One of the interesting hypotheses is to think about whether or not it's ACE2, some variant of ACE2, or if a history of exposure changes properties of the barrier tissue in and of itself to alter infection. And so with some support from one funder, we're now working with partners in Calcutta, in Bangladesh, in the Bahamas, in a couple countries in Africa and in Latin America to go characterize what nasal epithelium looks like in these different environments as a result of diet, environment, exposure, et cetera, to see if there might be something that would help us to get at that. I don't know that we're powered to think about the genetic associations, but we'll at least be able to think about whether or not there are changes in basal barrier properties that might be important.
And also the differences between the upper and the lower respiratory infections, or vis-a-vis different variants.
Yeah, I think it's a great question. For humans, it's really hard to sample. One of the nice things is that, by sampling in the nose, most people are willing to let you swab their nose. They don't like it, but we've become accustomed to it at this point in our lives, unfortunately. And so sorry, my dog wants to come in. She's howling right here. Maybe you can hear my husky. But we've become accustomed to it. On the other hand, sampling from lung is hard. In some places we got bowel samples, but bowel isn't really lung, and what you get is a lot of sloughed off dead cells that don't give you great data. In the non-human primates, we've done some things where we've looked at some of these pieces, but the genetics are hard there because the monkeys aren't inbred, and it's hard to get the variants that you might want to link at some of those relationships.
Rahul, can I have a quick, sorry.
Go ahead. Yes, Doctor.
Yeah, just quick comment and a question. So related to Dr. Shashidhara's question, maybe there are polymorphisms that actually change expression. I haven't followed that area that well. But if they do,
there
that
totally
would,
might be a shift, but we have to screen it. We just don't have power right now to look at like EQTLs in some of our data, and so it's a small number. We tried, as I said, we tried to look at whether or not there were mutations in interferon that were known to play a role there. But I think there's this broader question of how genetics and environment alter barrier to impact severity of infection that we'd love to look into, and we're starting to collaborate on with others.
Yeah, I mean, the genetics has worked out much stronger for oligoasynthase and some things like that, where we have big differences. I just also wondered, I mean, there is some data with Omicron now about it's not docking as well onto TMPRSS2. And that might be what people are getting at in terms of the differences in, upper and lower respiratory tract. Because you have more
Totally. And so we're going to go back and look, but the big thing that we want to do shift from our data is, because it's human and in vivo, is we want to figure out in the nose if there's a difference in the cells where we're detecting virus. We've aligned a virus, but we haven't done the deep dive as of yet. But the plan is to actually go in and say which cells in the nose have virus in Omicron versus in Delta versus in sort of the original strain. And use that as a way of understanding, whether or not there are specific things that associate with infection across them that might be putative receptors. And then we'll go back and look at that in lung as well.
Thanks very much.
And there are a couple of questions from some students. Class 1 molecules are involved. Is there any specific haplotype which is common amongst patients?
We just don't have power to think about that. This is not a well-designed cohort in that sense. It's well-designed in the sense that we have, a good number of mild, moderate, and severe, and that we have some, severe non-COVID controls. But we're not powered in those ways. We don't have the ability to think about, how gender or ethnicity or any of those things might play a role. And so I'm very reluctant to comment on those things just because it's 60 samples, but that's a very small. You should just think about links between genetics and this, given the things that were brought up before in terms of like differences in the slight timing of disease or the like. And so they're fantastic questions. They're things we'd love to study, but they're things that, we're just going to have to collect more data on, and where meta-analyses of all of the data sets that others are collecting as well might lead to some interesting insights.
Yeah, just one last question from another student. Is there any possible correlation between the reduction of interferon and COVID variants?
That's what we hope to see. We're hoping that what we can see is that there are differences in what we see across strains. One might imagine that there might be less, interferon induction in Delta given severity or something similar to that effect, and that in Omicron, actually, maybe another hypothesis, along with the one that Shiv raised, is that maybe actually you're getting stronger interferon responses in the epithelia. And so you can become more infected, but you help to maintain it locally as opposed to getting down to the lungs. So, not sure yet whether it's that there are fewer cells that are receptors for Omicron as you go down into the tissue, or if there are differences in the response of the nose where you're getting sort of initial infection that might play a role in helping to explain. It's probably some combination of both, because everything in biology is a combination of both, or our inability to tell the difference between the two.
Thank you, Alex. That was wonderful. Thank you so much.
Thank you so much for having me.
Okay, we'll move on to our next speaker, Dr. Nimesh Gupta. So Dr. Gupta received a PhD from Georgia University here in India and subsequently trained at INSERM and Institut Pasteur in Paris. Dr. Gupta's lab at NII works at the interface between basic and translational immunology, with a focus on flaviviruses and coronaviruses. By providing helper B cells, T follicular helper cells are crucial to the development of germinal center-derived antibodies, and the factors that govern the differentiation of such cells and their impact on the development of optimal humoral responses remains uncharacterized to a large extent. So Dr. Gupta's lab attempts to elucidate the traits that T follicular helper cells in long-term sustained immunity to help understand the characteristics of immunological memory. And his lab employs both human vaccination as well as natural infection models. And he has recently shown that the SARS-CoV-2 cross-reactive Cd4 T-cells exist in unexposed individuals, and has shown evidence for spike-targeted immune memory in mild COVID-19 patients several months after recovery. Dr. Gupta.
Thank you so much, Rahul. And thanks to the committee for this wonderful opportunity for sharing our work. So as we are, looking into the different aspects of particularly the T-cell responses in both infection and vaccines for COVID, I will just share some of the work that it's developing in case of the COVID vaccines. Particularly here, I will talk about the Indian inactivated virus vaccine and how it looks in terms of the immune responses. Hope you can see my slides.
Yeah, go ahead, Nimesh, go ahead.
Thanks. So I think we all agree that the vaccines have been tremendously helpful in at least controlling the disease severity, and I think this is the most efficient tool that we have in hand right now for the control response to this pandemic. And there has been mammoth efforts put forward in this past almost 2 years. And if you see the current landscape, we have almost 195 vaccines in the preclinical development. The 145 vaccines in clinical development, out of which 32 has either reached phase 3 or has already successfully completed the phase 3 trials. And almost 10 of them are now at the stage where we are looking into their kind of post-licensure phase 4 trial surveillance data. So if you see here, particularly we talk about these vaccines, there were several platforms that were trialed, that were tested. Several new platforms came up, and in this particular the groups, the two major components were targeted in the vaccines. One was the component vaccines, the virus component vaccines, and the second was the whole virus vaccines. Now, if you see in these virus component vaccines, majority of them, either they are subunit vaccine or the mRNA based or the vector based, they all actually target the spike protein of the SARS coronavirus 2. And for these whole virus, there are actually majorly the inactivated virus vaccines were made at the multiple the places in the globe, and there are few live attenuated vaccines that are now coming up. They are now reaching to the clinical trials, particularly the phase 3. One of them is now reaching. So if we talk about these vaccines, the most commonly used vaccine till today are particularly these types. When only one of them, which is a subunit vaccine, has reached to this phase 4. However, we have these mRNA vaccines and non-replicating vector-based vaccines, which are commonly used across the globe. The inactivated virus vaccines, particularly they've been traditionally developed utilizing the very existing technology, but some of these vaccines are of great interest, not just because they are using the whole virus, but they are also actually using a unique combination of adjuvants, which has never been trialed in a mass population. And if you compare the ongoing studies on all these vaccines that are available across the globe, we know least about at least these inactivated virus vaccines in terms of their capability of touching upon the immune responses, in terms of their capability of inducing the memory or kind of the breadth of protection they can produce. If we see some of these inactivated virus vaccines, I'm just listing here around these 9 candidates from the different countries. Almost this, like the CoronaVac or the Sinopharm, other vaccines are already has reached to the phase 4 now. They are in a surveillance stage in multiple countries. Majority of these vaccines are using alum as an adjuvant. And some of these vaccines, which are developed either here in India, which is this BVV152, or like in UK, they are using a combination of adjuvants. Particularly here, if you look into the vaccine which has been developed by the Indian pharma, they're actually using a combination of alum, which is actually absorbed with a TLR7/8 agonist, the imidazoquinoline. So particularly what happens with these, when there is no knowledge available, there were two, the major questions that were posed. First, do they really, are helping somewhere in providing the immune memory against SARS coronavirus 2? And the second, the larger question that will come on, as we start learning about these vaccines, about how these adjuvants are actually helping, whether they are making the difference in terms of bringing up a more protective or the breadth of protective immunity. So I'm just giving you, the bit of information on this particular vaccine, which has been rolled out in the country sometime in January 21. And this vaccine, of course, it is still the second the vaccine in the country, because the majority of the initial vaccination happened with the Covishield, which is the AstraZeneca the molecule. This vaccine, Covaxin, was developed utilizing, the virus which was isolated from a tourist on the D614G variant. It was inactivated utilizing a traditional method of a propiolactone. And the adjuvant which has been used here is this imidazoquinoline, which is adsorbed on alum. And that was mixed in this inactivated virus, which is around, in total around 6 microgram of the viral particles they use per dose. And it is, this vaccine is used in a 2-dose injections, which are given 4 weeks apart until today. What we know about this vaccine when it started, the rollout started, and on the way we started learning about the data that came out from the phase 2 or very recent, the phase 3 trial. What we learned that, after the dose 1, we see somewhere around 30 to 40% of the seroconversion that has been shown in this paper published from the Bharat Biotech. And as we move ahead, around 2 weeks after the second dose or 4 weeks after the second dose, the seroconversion leads to somewhere around 97 to 99% of the participants. And here they compared just the 2 doses from 3 microgram and the 6 microgram, which didn't so much difference. They also put on some of the data on the T-cell responses, and they show that, after 3 months of the second dose, there are some, inclination observed in case of the memory T-cells. Very recently, while we were also concluding our study on a very prelim information on the immune responses, the phase 3 trial data came out, and what they actually showed here, that this vaccine effectiveness in around 17,000 to 18,000 participants is almost 63% against asymptomatic infection, 77% against the symptomatic infection, and almost 93% against the severe infection. And at the same time, 1 or 2 other studies came out, particularly that has happened in the healthcare workers in a real world scenario. And what they found is that the effectiveness from the symptomatic infection in their cohort was somewhere around 55 to 57%. And they probably did not, account more on the severity index or the severity of the disease. But there are now more such, the information is coming on this vaccine. Something which, these studies failed to provide was more information on whether this vaccine is inducing an immune memory, and whether the memory responses are actually induced by this vaccine are capable of responding to the variants. So some of these questions and some of these information which were not available from this trial data, we tried to look into some of these questions. We tried to address these questions, particularly to look into the cellular or the humoral immune memory induced by this vaccine. Does it happen? And if it happens, how long it persists? That is still an ongoing question. And how these responses are actually responding to the recently circulating variants, particularly at that time when Omicron was not there, it was the Delta which was at the height. So to address these questions, what we did, we established a cohort, which was a one-time sampling cohort of around 71 participants in the group of vaccination. And these individuals were basically with the median age of 43. These individuals before vaccination and after vaccination until the sample collected were never, had any RT-PCR positivity, and they were also not having any kind of symptoms, which we confirmed on the basis of the routine follow-ups. The median days of sample collection for this particular cohort was 120 days, the median of around 4 months. And we also included a cohort of a mild disease patients, particularly those who recovered from the mild COVID-19, and tried to see that, what extent of responses this vaccine is actually inducing, and at what level it goes if we look into the case of natural infection. So the mild disease cohort was of around 73 individuals. The median age was around 33. All of them were tested positive by RT-PCR and confirmed for the mild symptoms on the basis of the WHO criteria. The only the status which is not very comparable with the vaccine cohort was the median of sample collection for this cohort reaches to somewhere around 7 months down the line. So what we did, we very first looked into the antibody responses from these individuals which were actually responding to this vaccine. And what we found here, if you see that the spike targeting antibodies were actually present in almost all these individuals, those who were vaccinated. The titer was not very different than what you see in case of the natural infection. The similar observation was made with the antibodies that are targeting the nucleoprotein. And here also we didn't see a major difference between the antibodies titer, particularly between the two groups. And they all were actually seroconverting here on the basis of what the cutoff that we made utilizing the pre-pandemic samples. Also, the responses that we observed here were actually well coordinated and well correlated between the two groups, particularly the nucleoprotein and the spike antibodies correlated very significantly, suggesting a very coordinated response against multiple proteins of the virus in this vaccine. And the similar, the observation was also, exist in case of the mild disease. We then looked into the neutralizing antibodies. So here, this is the initial data that we generated utilizing this microneutralization test. And what we observed that this test was done at somewhere around 1:10 dilution. So this is not a very great neutralizing potential that we can see, at least in this set of samples, where somewhere around 80%, 90% of them are showing the neutralization potential. But if we see and if we increase the titers, they will actually go down below the anti-50, below the 50% neutralization. Suggesting although they looks like in the same line like what you see with the mild disease, but their capacity of neutralization is not that great. So now we are actually looking into much more details with the pseudovirus setup and against multiple variants. And then we further looked into these antibodies, that how long they persist. That was particularly the more important question for us to see, particularly for the binding antibodies. And what we found is that if you see the spike IgGs or the nucleoprotein IgGs, they show a very good persistence, at least until 6 months of the sampling of these vaccines. And which was actually in line what we see in case of our cohort from the mild disease, with a very good persistence of almost a very high titer antibodies binding to both the spike and the nucleoprotein from the 6 month down the time to at least here in case of mild disease by like almost 10 months. So this information, what we actually accumulated on the basis of the antibodies suggested that the vaccine at least is inducing a good binding antibodies, but of course their potential in neutralization may be weak. The more details are soon coming out. But then we try to address whether these antibodies are actually able to respond, to the variants, and what level of reactivity they can actually provide when the different variants comes in. So here we utilize the RBD protein and the in-house ELISA, utilizing this RBD from the different variants, particularly from the alpha, beta, the delta, and the kappa and gamma, because kappa was actually circulating previously in India before the delta came in, and utilize these RBDs. And what we found is the two of these variants RBD, particularly the kappa as well as the gamma, they were actually not impacted by the antibody responses. So there's not much difference we observed between the RBDs of these two variants or the ancestral virus. However, we found that there is a decline in the capacity of these antibodies to recognize the RBD of the mutants like alpha, beta, or the delta. The most drastic, almost tenfold decline was observed in case of the beta, followed by delta and then the alpha. So particularly the response towards the beta was not something which is very surprising, because we have seen these responses against the beta, which is, I think, the most immune escape in terms of the antibody responses against multiple vaccines. However, in case of delta, we also found that there is almost a 7-fold decline, which is not as same as we can see in case of the recovered individuals. So these antibodies, which are at least the binding antibodies to RBD, they are also not able to have a very strong recognitions to the variants like delta or the beta. So then we try to address that, in case of the antibodies that we have seen, there is an impact by these variants, and the neutralizing potential looks like a bit weak. What happens to the memory responses? Because there was no information available on the memory B cells induced by this vaccine. So we tried to look into a very prelim information on the memory B cells by utilizing the setup of a polyclonal activation followed by the antigen specific analysis of these memory B cells, secreting IgG, IgM, or IgA, utilizing the ELISpot setup. What we found, I'm just sharing the data on the IgG antibody secreting cells here. What we found that almost 85% of the individuals are actually showing the existence of IgG positive antibody secreting cells. That's of course the range of these cells which is available in these individuals among the total IgG positive ASC cells is not very different than what you see in case of the COVID-19 recovery. But what we found is that some of these individuals who do not show the existence of the memory B cells, we thought that maybe something because of a time duration, a time lag in these individuals that might have led to the level where we are not able to detect them in circulation. So we just tried to look into the analysis between the frequency of these cells across the duration while we collected these samples. Surprisingly, it is something which is a very other way around that we found that the individuals, those who were actually sampled early after the second dose of their vaccine, are showing a very low frequency of these memory B cells in this cohort. However, as we move ahead from the time, we found a very good frequency available in these individuals. So this is something which we are still trying to figure out that what is the probable reason here. But at least these individuals, those who responded, are showing a very persistent IgG positive B cells in a long run, at least until the 6-month duration that we tested. And these B cells also correlated well to the RBD IgG that we tested initially. So with these analyses, we at least found that this vaccine is capable of inducing some memory, and the memory B cells are also induced, which are actually showing some persistent, and looks like they have enough capacity of inducing a lot of binding antibodies that are coming out and that are actually staying for a long term, at least until 6 months while we tested, and the long as we are moving on. Then we looked into these RBD binding B cells and just looked into their capacity of recognizing the different variants RBD. And like what we see in case of the antibodies, the similar trend was observed here, that for the COVID recovered samples, for the COVID recovered patients, there was much more decline that we observed against for the IgG positive ASC cells. However, that was not the same scenario in case of the vaccine. And we found that whether it's a beta or it's a delta, we have around 2-fold decline in the capacity of the RBD from this variant in recognizing these memory B cells. And similarly for the alpha, it was not a very huge difference which we observed, but of course there was a decline which was almost 1.5-fold. And the variants like gamma and kappa, they did not show much impact on the recognition like what we see in case of the antibodies. So we then thought that maybe an impact on the overall humoral compartment in terms of the vaccine-induced responses by these variants. So let's try to look into what happens to the T cells, because that information was also not very clear from the trial data. So we just set up and utilize these AIM-based setups to understand the frequency of the antigen-specific cells. And here is some data which I'm sharing that if you see here in this second panel, which is showing you the spike-specific Cd4 T cells, we found that somewhere around 85% of the individuals are showing the detectable T cells in the vaccine group. And which we also found was comparable to to the frequency of these antigen-specific T cells in case of the recovery from the mild disease. And when we looked into the capacity of these T cells in recognizing the variants, particularly we focused here on only the 3 variants, alpha, beta, and delta, we didn't see a huge impact of these variants on the existing T cells that came out from these vaccine responses. Only in case of the delta, if you see, there was a decline in somewhere around 10 to 12% of the individuals, those who actually lost the capacity to respond to the variant pools. However, if we see an overall median response, that was not much impacted, both in case of the vaccine as well as in case of the infection. We further then looked into the memory compartments, because these cells which are showing the frequency, we didn't know what kind of compartment they are actually coming from. And what we found is that both the central and the effector memory compartments are enriched from these vaccine-specific CD4 T cells, and which are actually similar to the compartments what you see in case of the natural infection. And they actually show a very good persistent here. If you see in this particular plot, across the time from away from the vaccination, the central memory compartment, which is in the sand color, is actually showing a good persistence in these individuals, suggesting that it has actually populated in both the effector and the central memory compartment, and it is actually persisting at least up to the 6 months post-vaccination. We further looked into these cells in terms of, do they have some kind of a functionality for either the antiviral or the kind of B cell health functions. So we looked into their capacity of secreting interferon gamma and other multifunctional capacities. I'll just show you this data, which is talking about their multifunctional potential in terms of secreting the cytokines like TNF, IL-2, interferon gamma, or the other cytokines like 17, 13, or IL-4. So what we found is they have at least the cytokines coming from the Th2 kind of cells like IL-4 and 13. However, they are more enriched in case with the capacity of secreting interferon gamma or IL-2. Something which was more surprising for us is that a high load of TNF alpha, which is actually coming out of these cells, which is of course comparable, at least in this cohort, with what we see from the mild recovery patients. But still, that was one of the highest cytokine that we have seen in these cultures. So though it gives us an idea about that there is a Th1 kind of polarization in these particular subsets, but it also gave us a kind of a hint that there is something which is more a kind of TNF secretion happening from these cells. So because we would like to look into definitely into the more on the TFH cells, whether this vaccine is capable of inducing the TFH cells or not. Like what we have seen in case of the other vaccines, other the mRNA vaccines, which has been largely studied in the last year. But we were also trying to understand that whether these cells which are coming out from the vaccine responses, is there something which is making them not very functional if they are having a lot of TNF alpha, which we were just trying to correlate with the study which was published by Dr. Pillai and group, that there were a lot of TNF alpha secreting cells exist in a scenario where these GCs were not formed, and of course the TFH differentiation did not happen. So we then looked into the overall CXCR5 positive cells either expressing PD1 or not in response to this vaccine, and what we found is the vaccine is inducing the TFH cells. These are just the spike-specific TFH cells, and they are, of course, if we compare with the COVID-19 recovery, they are not, they are not as high as in case of the infection, but they still, have a fraction of TFH cells that exist there. And when the TFH cells are also there in case of the nucleoprotein, which is very comparable with what we see in case of the infection. We further looked into their heterogeneity compartments, and unlike what we see with the Th1 subsets in case of the Th population, here we found that these TFH cells are actually existing with both the phenotypes. They have a TFH2 and TFH1 phenotype, which is actually also reflected in case of the recovery, but not as prominent as we can see in case of the vaccine. And the NP specificity of this TFH do not show the similar kind of profile what we see in case of the spike. So this was just a kind of frequency that we see, and then we tried to understand that whether looking into these cells and looking into the previous data on a lot of TNF alpha availability and secreted by these cells, is there an effect on the functions of these the T cells? Particularly the functions that we are interested are the B cell helping function. So there were the different set of different assays available till date, which were actually looked into the B cell help potential of the CD4 cells, but none of these setups were able to look into the antigen specific responses because of probably the poor sensitivity of these cultures. So Asghar in lab has been working on these setups to try to generate a way where we can go into a very sensitive the method which can allow us to look into the antigen specific T and B cell interaction even with a very low number of cells. So he has found some of those factors recently, which I'm not discussing here, which can actually help in improving the sensitivity of these T and B co-cultures in the ex vivo conditions. And he has even reached to a level where somewhere around 90 antigen specific T cells can be analyzed for their capability of providing B cell help in the ex vivo conditions. So he quickly applied this setup and asked the questions whether these cells, what we are seeing in case of the vaccine, are they having the B cell help functions, which is of course very important for the humoral immunity establishment, equivalent to what we see in case of natural infection or not. So he took this setup and took the cells from a cohort of around 20-24 individuals from both the groups, and he found that like what we see in case of TFH, the capacity of these cells in secreting IL-21 is again low as we see as compared to the COVID infection. However, if we look into their capacity of providing help signals to the B cells and get them differentiate towards the plasma cell is not very different. So they still, though they are less in number, they have the capacity to provide help signals to the B cells for the further establishment of the humoral responses. So with this, the current the data, what I can summarize here that this vaccine looks like having a very persistent spike in NP binding antibodies, at least up to the 6 months. We do see that the RBD specific antibodies drop in the range of 3 to 10-fold against the different variants ranging from alpha, beta, and delta. The vaccine is capable of inducing the persistent memory B cells and also inducing the antigen specific functionally potent T cells in almost 85% of the subjects, and these T cells are largely preserved against the VOCs. And more interestingly, it has the capacity to actually induce the adequate quality TFH cells, and more, of course, need to be now learned from these TFH cells in a long term, how they are responding. Just a quick takeaway from what we believe that this vaccine have probably a low tendency in terms of inducing the neutralizing antibodies, but it seems to have more strong T cell responses, which might have a large breadth of reactivity against the variants. And maybe a booster immunization, either a systemic or intranasal, may be helpful in increasing the breadth of these humoral immunity. So with this, these are the guys, those who have, worked throughout this a tough time. We were a new lab. We sailed into COVID-19, and because of their enthusiasm and a very strong interest and passion for these the T-cell work, Rajesh and Asghar were particularly involved in this T-cell work, and Someshwar, Anupama, and Bhushan, they have contributed in more of the B-cell side, and also now developing the pseudovirus platform for our studies. And Anurag has now joined in NIH for her postdoc. She was my first student from the lab. And these are the collaborators, those who have contributed a lot in this study, and of course on the ongoing program from THSTI, from All India Institute of Medical Sciences, ESIC and LNGFP hospitals for their continuous collaborations on COVID-19. We have been on a huge programs going on with Alex in both COVID as well as in some of the flavivirus programs, and Alan and Etienne from MRC and IPSB Toulouse for their contribution in setting up these HET assays in our laboratory. And with this, I thanks all of you for your attention, and I'm ready to take your questions.
Thank you, Nimesh. Shiv has a question.
Yeah.
Dr. Pillai? Yes.
Yeah.
Yes, please.
All right. So Nimesh, that was really interesting. Very nice work going on there. And I also really like what you guys are doing with the in vitro stuff. There is a semantic issue in the field that I just want to clarify. So I don't think in COVID there's any shortage. So what we see in the circulation sometimes gets loosely referred to as circulating TFH and so on. But there's never any BCL6 expression. Similarly, if you look at a lymph node, the majority of the cells which are CXCR5 positive, either in the mouse or the human, much more prominently in humans, are outside the germinal center. They don't have BCL6. So there are plenty of cells that provide T-cell help to B cells. These don't make germinal centers necessarily, unless they are BCL6 positive. So some of them are TPH cells, what Deepak Rao and Mike Brenner have been saying. Others are T helper cells with CXCR5, and often with PD1. So there's some differences. So I think what you see in the blood is really what we would call pre-GC. I mean, I know that lots of people in the US also call them circulating TFH, but just to make that clear, there is no real defect in those cells, even in COVID. It's only the BCL6 positive. For that, you have to look at a lymph node. And I mean, some people in vaccination have done those, needle aspirates. But just to clarify, I mean, I understand where you're coming from on that statement, but there's something to keep in mind.
Yeah. Thanks so much. We'll go into that, because we have also seen some of the cells which are expressing PD1 but not CXCR5, and they have a very strong potential of providing help to B cells, and that has now been seen in case of dengue as well. So I think there is much more than what we see and only believing on TFH providing the help, but I think more will come soon on
those cells are what Deepak published in his paper, STPH cells. Yeah, they all have similarities. Yeah.
Okay, is there anybody else who wants to ask a question? Yeah, I don't think there's anything. So, Nimesh, I was just wondering whether the neutralization varies as time goes on to different variants in immunized versus, I don't know whether you presented, I may have skipped it. If it varies in the immunization versus sick people of the variants. The neutralization of the variants varies as the time goes on. Does it stay the same, or the ratios are different?
So if we talk about the neutralization, the neutralizing antibodies against the variants, we do not know until now. We are still, that data is we are collecting right now, and only the potential of those antibodies initially that we have seen, we didn't see much difference in terms of the vaccine or the infection. But right now, I cannot exactly answer the question what you are asking. But yeah, soon, maybe in a couple of weeks, we'll have that answer.
And also, you mentioned about multifunctional or polyfunctional T cells. You don't know whether the same cells are making all the cytokines, because it's a co-culture, right? So you don't know which cell is making which cytokine.
Yeah, that we have done at both the bases, in the co-cultures, as well as we also looked into the ICS, and using the ICS, where you can do kind of Boolean gatings and identify the cell types which are actually secreting. So we found the scenario where a lot of TNF alpha secreting cells are much more enriched here than the 3 cytokines or the 2 cytokines like interferon or IL-2. So still, we do not know why there is a lot of TNF alpha secretion, which is much more unique here in case of these cells, more than interferon, IL-2 and all. But yeah, there is something needs to be looked into in the future.
Okay, thanks. Someone has a question.
Hey, Nimesh, nice talk, and nice to see the way you were characterizing this vaccine response and vis-a-vis virus-infected individuals. Curiosity is that this is an inactivated virus vaccine. And when I looked into data, this is true that there was more TNF expression, and there is a inverse relationship, I couldn't get it, that TNF and interferon gamma, expression of TNF and interferon gamma, they were, one was up in virus infected individual vis-a-vis vaccinated, and interferon gamma was down. Any thought on this? What could be triggering it? I mean, one is live virus, another is inactivated virus.
Yeah, only thing, what we are seeing is particularly this inactivated vaccine, that interferon gamma responses are not that great, if we even compare with, because this is something which is with the mean responses that we looked into. They are not very different between the infection and the vaccination, but interferon gamma responses are not that high. But yes, the TNF alpha responses are quite high. We think that it may be something which is coming out because of the adjuvant, but of course at this stage we cannot really clarify that particular question. But in future we will try to see if that adjuvant is something which is leading to that trait of the cells.
No, but the one line of debate would be that adjuvant did not lead to interferon gamma upregulation, right? Just, I mean, as a thought, of course. Yeah,
maybe possible.
All right, thank you very much.
Thanks.
Thank you, Nimesh. Thank you so much. We'll move on.
So much.
Yeah. Dr. Gaurav Gaya is here?
Yes.
He's here. Excellent. So he's next, and Dr. Gaia received a DPhil in biochemistry at the Oxford University and MD from Harvard Medical School and MIT in the Health Sciences and Technology program. He subsequently trained in internal medicine and gastroenterology at the Massachusetts General Hospital. So there is currently a renewed focus on the development of broadly protective T-cell based vaccines. And as a research fellow at the Reagan Institute, Dr. Gaia determined the significance of T-cell targeting structurally constrained regions in the treatment-free control of HIV. And in his current work has led to the identification of CD8 T-cell epitopes across the SARS-CoV-2 proteome, which impair infectivity when mutated. So he's going after such epitopes, and such epitopes which elicit CD8 T-cell reactivity in convalescent individuals may form the basis of a global T-cell vaccine in the future, and the efficacy of which may remain unhindered even upon the emergence of new variants. So Dr. Gaia, please.
Well, thank you very much for this opportunity to speak and to share our work. It's really a privilege. I'm just going to mention that both my parents grew up in New Delhi, so they were particularly proud of this invitation for me. So it's wonderful to be able to share my work at this venue. I'm just going to give a brief overview of just the seminar for today, which is really going to be composed of two sections. So one is really more of a basic immunology study that we just completed and was published, looking at T-cell responses, particularly to the Omicron variant, and I think some new emerging lessons that we gained from that study. And then the second is related to the work that was just described, where really taking from some of these lessons, how we could develop a variant-resistant T-cell vaccine. And this is kind of more of a technology talk, and will relate to a technique that we've developed in the lab initially for HIV, known as structure-based network analysis, and how we applied it to SARS-CoV-2. So the vaccines for SARS-CoV-2 have really been quite a revelation, really a miracle of modern medicine. And they seem to work primarily by inducing protective antibodies by delivering the spike protein in various formats. Also, you have the inactivated viruses, which seem to be driving antibodies as well. But I think we've seen over the past year that the waning titers and reduced neutralization of variants of concern do raise some issues regarding long-term protection from the current vaccines. And so when we think about antibodies, we have these issues with durability, with kind of these waning responses that have been seen about 6 months after vaccination, that reduce, that waning in titer also leads to reduction in vaccine efficacy that declines with time. We're also now seeing partial escape from some of the variants of concern, particularly beta and delta initially, and then obviously with Omicron. And in part, that's due to this targeting of the spike protein to mediate protective effect, where a lot of these mutations are rich within the RBD and the S1 domain, where neutralization is primarily mediated. I think what that raises is that the other side of the adaptive immune response, particularly T cells, and I would say even more specifically CD8 T cells, should be leveraged to gain additional control of this virus, and particularly to mitigate against moderate and severe SARS-CoV-2 disease. So first, CD8 T cells are very long-lasting. They're quite durable. So we saw this in COVID-1 infection. We also seen that in individuals post-COVID. In terms of outcomes, they're really associated with mild disease, abortive infection, and can even mediate protection in individuals who lack antibodies. They have far greater preserved recognition of the variants. And that's really because they can mediate their protective effect by targeting the entire expanse of proteins. So whether it's the full spike protein, or in the case of recovered disease, some of the non-spike proteins, and you can also see that likely in the inactivated vaccines as well. So just greater landscape with which to target the virus and mediate a protective effect. So the Omicron variant, really came to worldwide attention late last year. And there were a number of studies that were very rapidly done. This is just one from the Reagan Institute that was done, which really demonstrated this reduced neutralization by antibodies of the Omicron variant. So very nice work by Alex Balazs, which I'm sure he shared yesterday, where you do really see a reduction in comparison of neutralizing capacity of wild type versus Omicron in a range of patients with different phenotype, whether it's recent vaccination or prior infection vaccination. And that's largely in part because of this enrichment of mutations present in kind of key sites where antibodies will mediate protective effect if you compare it to either Delta or the wild type. The question we had was what effect would these mutations that seem to be largely enriched in the spike protein and then somewhat in other proteins of the virus, how would that affect CD4 and CD8 T cells that are induced by prior infection or vaccination? And additionally, in the US, we've had this rollout of booster vaccines. What effect would booster vaccine doses, how would those affect T cell responses to Omicron as well? And I will say that, because in spike, there really were only about 3% of the residues mutated, our hypothesis was that we would expect very limited effect of these mutations on CD4 and CD8 T cell recognition. So we put together a cohort of individuals from Chelsea, Massachusetts, which is adjacent to Boston. These individuals had a range of clinical phenotypes. So they either had no prior infection or prior infection, were vaccinated, or unvaccinated or vaccinated with the vaccines that had been distributed in the US. And then there were a subset of individuals who further had booster vaccines as well. We had 76 volunteers in total. And then here are just the additional numbers in terms of those that had those different clinical phenotypes. And then the time of collection. Generally, it was about 6 months after primary vaccination series that we were able to obtain samples. And then post-booster, really, it was very short term after the boosters, a median about 10 days with as much as almost about 2 months that we were able to collect samples. We essentially decided to look at T cell responses to a range of different peptide pools. Of course, the Omicron spike, and this is consistent with about 5 or 6 studies now that have come out. But we also looked at the Omicron structural non-spike proteins, so nucleocapsid membrane envelope, and then also ORF3A. I think we were the only study to really look at this additional peptide pool to look at the non-spike antigens. And we used very traditional T-cell approaches that we've used many years within our HIV studies, looking at an interferon gamma ELISpot just to capture the total effector T-cell response, and then a CFSC-based proliferation assay to look at both CD4 and CD8 T-cell memory, but also functional T-cell responses. So here's just some raw data that we were able to generate, and this is just the setup. So here's an individual with no prior infection, no vaccination. Just our positive control, seeing that clearly our assay is working, and then exposure to the different peptide pools. We see basically very limited underlying cross-reactivity of these peptide pools. And then we looked at individuals with either prior infection, vaccination, or hybrid immunity. And what we found was, in most people, this kind of beautiful preservation of responses. So you saw similar reactivity to T cells to either wild type or to Omicron, which was similar to Delta in these individuals. And again, this was the vast majority. But what was striking was we also found a subset of individuals who lost reactivity to Omicron, who did not have preservation. And here are 3 of those individuals located here, is shown here. Here's individuals with prior infection with a very robust T-cell responses spike that really had a complete drop-off or loss of their T-cell reactivity to Omicron. We see that in a vaccinated individual, and then an individual also with hybrid immunity. And so this indicated, and we saw this in about 20% of people, really that there's a capacity for Omicron, despite this low percentage of actual mutations within the spike, to actually have a very profound effect on T-cell reactivity in some individuals. So here's the composite data looking at these 3 groups, and really the ones with these red lines are those that had at least a 50% reduction in their T-cell reactivity by comparing what they had for wild type versus Omicron. And this was about 21% of individuals in our cohort. If you compare that to Delta, we really only saw about 9% of individuals with a reduction in the T-cell response. So it does seem that, with this continued evolution of the virus, we are seeing this loss of neutralizing antibodies. We're also seeing an increasing reduction in T-cell reactivity from those that were induced either with prior infection or with vaccination. What's interesting, though, is you actually look at the non-spike proteins, we really had no individuals who really had any reduction in T-cell response. This is certainly due to the lower level of mutations or mutational burden within Omicron in these proteins. I think this highlights that these represent potentially very valuable targets for vaccines. They're definitely incorporated in the inactivated vaccines, but as we've seen, most vaccines seem to be very spike-centric. And so I think this data starts to indicate they're really incorporating additional proteins could be valuable for kind of future vaccine design or just as a consideration for vaccines moving forward. So what affected the booster vaccination on T-cell reactivity to the Omicron variant? Well, I'll just say that we were able to take pre and post booster specimens from about 25 individuals in this cohort. You can see that generally pre and post boost, there was a really rapid and quite robust increase in the T-cell response. Here are two individuals, one that was just vaccinated, one with hybrid immunity. You can see the robust activation of the spike-specific T-cells. And really, we saw about 20-fold increase overall within our cohort as a consequence of the booster vaccination. But what's interesting, though, is that still there were some individuals, despite boosting, who maintained this relative difference in the actual reactivity between wild type and Omicron. So here are two such individuals, either again a vaccinated or an individual with hybrid immunity, who still maintain a reduced reactivity to Omicron relative to wild type despite boosting. So I know there's been indication that you have this broadening potentially of the antibody response as a consequence of boosting. We, at least in some individuals, did not see that broadening necessarily of the T-cell response. And again, levels of T-cell reactivity maintain high, but again, there were about 10% of individuals who had still had a greater than 50% reduction in their T-cell response to Omicron, even despite the booster. And so this is data actually just correlating now the actual T-cell responses, which are on the x-axis, with pseudotype neutralization assay data from these same individuals. Generally, we saw a good correlation between those two, where overall effector T-cell response did seem to correlate with neutralization capacity. And really, with the two groups here, those are vaccinated and those with hybrid immunity down below. Well, what's striking, though, is we did see now this group and subset of individuals who did appear to have really no neutralization capacity and really no T-cell reactivity either. And this would obviously be a concerning group, where even despite vaccination, they really didn't seem to have any potential protective immunity against the new Omicron variant. So it does suggest that this further evolution of this particular variant is now potentially putting a subset of individuals at risk really for infection without any kind of protective immunity for this particular virus. So we also performed a CFSC-based proliferation assay, which really helps identify functional antigen-specific CD4 T cells. The way we do this is we just take PBMCs, we label them with this dye CFSC. When the cells divide, the daughter cells will take half the dye, so they actually will lose fluorescence. And then we actually take these labeled cells and just incubate them with different overlapping peptide pools. And here's just some representative data where you can see the cells start out very bright on the CFSC axis, and then as they divide, they actually end up shifting to the left as they lose fluorescence and move that way along the axis. So here's looking at the Cd4 T-cell responses. So now these are individuals who are either vaccinated with hybrid immunity or who actually were post-boost. And this is relative to the wild type spike or the Omicron spikes. You can see some very robust proliferative T-cell responses suggesting strong immune memory in these individuals. And this seems to be largely maintained. And so when we look at the comprehensive data, we see that despite a few individuals, generally memory CD4 T-cell responses seem to be maintained to Omicron. But really the story seemed to be different when we looked more specifically at the CD8 T-cell responses. So now we're here two individuals where we had, in this case, a 4-fold reduction in the memory T-cell reactivity to Omicron. Here was a 3-fold. Generally, the vaccinated individuals who received mRNA vaccines, we see predominantly in our cohort didn't have quite robust CD8 T-cell memory. So I know that is different from what some others have seen using different assays, but this has been a consistent finding that we've observed among vaccinated individuals. And so now what we actually found was that there's quite a substantial number of people in our cohort who actually had this reduced CD8 T-cell memory reactivity or response to the Omicron spike protein. And this obviously is somewhat of a concern. I will say that this finding, which was surprising to us and did seem to contradict some studies that have been put out recently, was very comparable to this one put out by a group in South Africa. So they looked at individuals from Cape Town and KwaZulu-Natal using an ICS assay. They similarly found that there was individuals with a complete drop-off in their CD8 T-cell response when comparing Omicron to ancestral. So this does seem to be somewhat of a common phenomenon, both common here in the US with our town outside of Boston, but also in individuals in South Africa as well. So the question was, what could be going on? And And our thought was, perhaps that this was due really to the specific epitopes that are changed and HLA associations. And so we just looked comprehensively at the mutations, and we looked at predictions of all the possible epitopes that could bind a broad expanse of HLAs in order to kind of really think about this from a global perspective. So that really allowed for prediction of about 736 unique epitopes. Generally, what we found was that about 94% of those epitopes did retain binding to HLA class 1, which is really the easiest mechanism for escape for a viral epitope to evade T-cell recognition. But we did see an emergence of about 7% of epitopes that did actually lose binding entirely to HLA class 1 molecules. So really, despite this small subset of mutations in the spike protein, there did seem to be this particular effect on epitopes on their capacity for T-cell recognition. And this is just looking at HLA binding. We don't know actually what the impact of those epitopes, of those mutations would have on, say, T-cell recognition or processing as well. And here are just some specific epitopes. We picked these out because these are actually different HLA alleles that were present within the patients in our cohort who lost reactivity. So they were enriched for HLA alleles. They had at least 3, and 60% had 4 to 6 of their A, B, and C alleles were actually potentially susceptible to mutations that were present in the Omicron spike protein in terms of what effect this mutation would have on epitope presentation. And therefore, that seemed to be a potential mechanism by which this decreased reactivity was taking place. So just a summary of this particular story, it seems that generally T-cell responses are preserved to Omicron spike in about 80% of the individuals, and definitely to the non-spike proteins. But as I said, about 20% with prior infection or vaccination had a greater than 50% reduction in their overall T-cell reactivity to the Omicron spike. There seemed to be reduced functional memory T-cell responses that were observed primarily in the CD8 T-cell compartment, with about 6.7% of epitopes found to lose capacity to HLA binding, again by prediction. And then just generally, booster vaccination certainly enhances the T-cell responses to Omicron spike and potentially brings it back to a level where it can be protective. I think overall, though, what this is saying, though, is that as the virus seems to continue to mutate and really distinguish itself from the initial ancestral strain, the question is, will this concurrent immunity that we have become less and less effective, particularly the vaccine-induced immunity? And is there ways we could really preempt this or get ahead of the emergence of future variants? And that's really the kind of the focus of this second part, which is how can we potentially develop a variant-resistant T-cell vaccine that could really mitigate against any future variant going forward? And that was really the kind of the goal from the beginning of the pandemic. And this really, again, leverages this technology that we developed for HIV known as structure-based network analysis. So this was a goal we really had back in 2020, which was, our focus being on T-cell vaccines was, could we design an effective SARS-CoV-2 vaccine that was composed of mutation-resistant T-cell epitopes that provide broad coverage of the global population? We really saw this in 3 steps. So the first was to identify mutation-resistant regions, again, using this network analysis technology. The second was to define T-cell epitopes in these regions that were presented by major HLA supertypes. So I'll touch on a new approach we developed in the lab, which is the HLA peptide stability assay. And then lastly was to evaluate the immunogenicity of these epitopes in convalescent and vaccinated patients, again, just by interferon gamma alpha, to confirm that these epitopes that were mutationally constrained could actually elicit T-cell responses. So for this first part, this really just builds off of many years of HIV, where traditionally, the way that epitopes had been evaluated was purely from a sequence-based perspective. It was really to look at T-cell epitopes in a linear context across thousands and thousands of viral sequences and try to identify those that they were conserved or non-conserved. And I'll say over many years, what's been found is that these conserved epitopes have not been effective in getting you broad protection against the virus. And that's really in part because when you look in this top-down way at sequence conservation, you're really not taking into account T-cell pressure, which is highly variable across people and in populations. And really the goal is not to be conserved as a residue. We really want to be going after residues that are constrained, that even if you applied pressure on those residues, they could not mutate. And to be able to define those constrained residues, we developed a structure-based approach, which really tends to evaluate the T-cell epitopes within the context of their native 3-dimensional structure. So rather this linear context that we see here, it was to look at the context of epitopes within its folded protein. And if it has key contacts and incorporates key structural constraints, that is really where you'd be wanting to place immune pressure on the virus, because any mutation that would take place in this linear context would then have an impact on the actual viral protein itself. So I looked at the literature to be able to do this and define structural tools that could give me insights into structural constraints. What I found was that you could look at things in very fine detail. So for example, this arginine side chain, you could look at these side chain interactions in fine detail, but I didn't know what the impact of this given residue would have on the overall architecture of the protein. And so to be able to have that new insight, the kind of jump in logic and the technology was really to try to represent this protein as a network. And so instead, the residues would become nodes and the inter-residue interactions would become edges. And then now we could use the power of network theory to really define amino acids or nodes that were critical to the network, and therefore if they were mutated and changed would have a very profound impact on the overall topology of the protein structure. So I'll just kind of go down just for a second, a little more detail. So here's our protein schematic. And you can look at it. Now these are residue inter-residue interactions. Already intuitively, you can see, for example, that this yellow node or this amino acid or these green ones are going to be very critical to the network. You mutate them or you change them, it's going to have a very profound effect on the structure. And just to make it a little bit more granular, you can imagine, for example, this is an alpha helix, this is an alpha helix, and here's a bridging residue interaction. So by using network theory, we can now define quantitatively those nodes that are critical or those amino acids that are critical. And we now call these particular residues highly networked amino acids. So I'll just say that we validated this on several proteins where we had lots of mutational data, like high throughput mutational data, and then we applied it to HIV. So here, for example, for the GAG24 protein, here's the crystal structure. We can now define the network by looking at all the inter-residue interactions, and this is really the composite of different bonds, from hydrogen bonds to pi-pi interactions, pi-cation, salt bridges, etc. And now we can now define quantitative scores, and really the score is defined by the size of the actual node that's present here in the structure. And furthermore, once we had individual residue scores, we could then assign TTL epitope scores. And we could find that there were very protective epitopes present in largely core regions or interface regions. And this is where individuals who were able to successfully control HIV were really targeting the virus. And then you had a lot of epitopes that were associated with risk or more rapid progression, and they were really present on more surface regions where any mutation would have very little consequence to the overall viral protein structure. So as a concept, you really want to be trying targeting viruses in these key regions and to apply maximal immune pressure, and really not at epitopes that are located in more peripheral regions of proteins. And I'll just say this is a game we play in the US, which is called Jenga, which is like this big tower. I played this with my daughters. And if you actually say remove a block, you'll lead to collapse. So this is an example of what a highly networked epitope would be, and one that's more peripherally would have very little impact. That would have no consequence on the protein structure. So we apply this to SARS-CoV-2. So now it's here to the SARS-CoV-2 spike. So here's a cryo-EM structure, and then now you have the network. Now we're able to define specific residues within the SARS-CoV-2 spike that are really constrained from mutation. These were generally at the interface of the trimers. We also found them in the hinge region between the S1 domain and the RBD. And other kind of core regions that seem to be really resistant to mutation. So we were able to do this for about 44% of the SARS-CoV-2 proteome. So here it's for some of the structural proteins, and then here are some of the non-structural proteins. And we were able to generate these scores very rapidly. So this again was about May 2020 when this was able to be done. So first we compared just the actual network scores with viral sequence entropy for different viruses. So here it is for SARS-CoV-2. We saw the highly networked residues here located in blue had very little viral sequence entropy, which is what you would expect. But what was really interesting is we saw the same thing for SARS-CoV-1 and also bat beta coronaviruses and also MERS, suggesting that these residues had a degree of structural conservation across SARS-CoV viruses and other lethal coronaviruses. Furthermore, when we actually look at them across the expanse of the SARS-CoV-2 proteome, so the ones here in blue are actually the highly networked residues. What we found was that back in May 2020, there was very little viral sequence evolution that had taken place, but Now, over the course of the pandemic, so this is in February 2021, about a year later, we saw that all the regions of new variation typically took place in residues that were poorly networked, and the residues that were networked continued to remain sequence conserved. And I will say I don't have the data here, but if we've looked more recently, we still see that these residues remain sequence conserved even as the viruses continue to evolve. And this was also relevant to the variants of concern, where really the mutations that were taking place in the VOCs was happening in non-network regions. And again, as I mentioned, we see that these same residues remain very highly conserved from sequence entropy standpoint across the sarbecovirus subgenus and then also MERS as well. So potentially giving us the capacity to generate a kind of pan beta coronavirus T-cell vaccine. So we want to evaluate some of these residues using the lentiviral pseudotype infectivity assay. I'm sure that Dr. Balas yesterday showed this assay, but essentially if you give a wild type spike in a lentitype pseudovirus, deliver this to 293A2 cells, you can get very robust infection, which can be detected by ZS-Green. So we actually took pairs of residues that either had high network scores or low network scores. All of these residues were sequence conserved, and they had the same identity, but then they were actually mutate the exact same residue. And these are all within spike. And we said, let's compare the impact of mutations within the pseudotype lentiviral infectivity assay. And what we found was really a profound difference, where if you took, for example, in this case, a phenylalanine that was networked and mutated to an alanine, you had completely ablated infectivity. If you took a residue that was non-networked and gave this highly non-conserved mutation, which we would expect to give some impact, still was robustly infectious. And so you had this striking difference that despite these residues being sequence conserved, all of them really being networked and sequence conserved was really consistent with having a profound effect on pseudotype lentiviral infectivity. So really, these are the constrained residues I was alluding to before. And we also looked at data from Jesse Bloom's lab, where he had mutated every residue in the receptor binding domain and then looked at the effect of either folding or ACE2 binding. I'm just going to show the data for folding. But we took our network score on the x-axis and actually correlated with his mutational data, where we averaged the effect on RBD folding. And we had this very striking inverse correlation with more highly networked residues having very profound effect on RBD folding. So really having some consistency with in terms of the overall predictions of these scores. The next was we want to define T-cell epitopes in these regions. The way we did that is by actually utilizing the HLA peptide stability assay. This makes use of TAP deficient HLA cells. So these express a single HLA allele, and then, but they're TAP deficient, so there's actually no HLA present on the cell surface. We take these cells, we can incubate them at 26 degrees and incubate them with a candidate peptide. When they actually are in 26 degrees, we'll have peptide receptive molecules come to the surface, and the peptide can actually then stabilize that molecule, is able to bind. Then you put it back at 37, anything that's non-bound will be re-endocytosed, and anything that can stably bind can be detected by flow cytometry. So here's just an example of that data. So for a known HIV immunodominant epitope, as you increase the concentration of immunodominant epitope, you get increasing stabilization of the HLA molecule as detected by flow. Using a relevant epitope, you get actually no stabilization. So at the start of the pandemic, this was available for just one HLA allele, which was HLA-A02. So during the pandemic, the lab generated 17 additional cell lines, and this was able to give us greater than 99% coverage of the global population. And so we then, for example, we did this for HLA-A03, and these are the predicted epitopes that would have binding. And we found that actually the predictions did not actually play out exactly with the experimental assays. So having this experimental assay allowed us to really refine selection. And so here we were able to see a number, in this case 5, that were predicted to bind that really had high stabilizing capacity. And this is relative to an immunodominant HIV epitope, which was always our comparator. And we saw that those that we predicted had at least 50% stabilization could be very promising COVID-19 vaccine candidates. So we did this comprehensively across these 18 HLA alleles. They gave us very broad coverage of the global population. The ones in dark blue are the ones that had at least 50% stabilization to the immunodominant HIV epitope that correlated with that particular allele, shown in red. And so from that, we took a set of about over 300 epitopes that were predicted to bind, and it could further really narrow down on 109 that were truly very good stabilizers of these HLA alleles. We found that 56 were from non-structural proteins and 53 were from structural proteins. So just again, looking at the location, they were kind of in these either core regions, this is an NSP16, or in these interface regions. These are critical structural regions, again, trying to hit the virus in areas that are going to be difficult to mutate. And when we look at its alignment with other coronaviruses, again, we see very strong conservation around the sarcovirus and to some degree to MERS as well. So lastly, we just want to evaluate the immunogenicity of these epitopes. And we've at that point looked at convalescent individuals. So we put together a cohort of individuals who have been infected with the ancestral strain. And we decided to look at, again, the reactivity, and here are all the different regions that we identify with that contained good stabilizing epitopes are here shown in green for the non-structural proteins and in purple for the structural proteins. And when we looked at our Cd4 reactivity, so here's a healthy donor, and what we saw was, again, no reactivity to the epitope, which was what we expected, but in the COVID-19 infected patients, we saw the most robust reactivity really to those derived from structural proteins, which is consistent with what we know now. That it does seem that the structural proteins seem to be the more immunogenic than the non-structural. And that really played out where we saw the most robust difference between the healthy donors and the COVID-19 patients was to the highly networked peptides derived from structural proteins. And so these really seem like the most promising to potentially make a variant-resistant T-cell vaccine, highly stabilizing HLA alleles and also immunogenic. And so here is just listed the actual different regions that we have identified. There's certainly a number that are located in spike. I think part of the issue with delivering the full spike and we discuss is, and in the current formats is that something about the kinetics and also all the other distracting epitopes. Really, I think having more focus on these regions in spike can really get this preferential targeting, but also including these other regions in nucleocapsid, membrane, ORF3A could be highly beneficial for both a broad and variant-resistant T-cell response. And so that's really, I think, where we're trying to move next is, the spike vaccines have really been quite a revelation, but I think augmenting those spike vaccines with a T-cell vaccine composed of networked spike and non-spike regions could be very beneficial. And as we saw with Omicron, this decreased reactivity that we're seeing, we could really prevent that and preempt it by incorporating a vaccine targeting these variant-resistant sites. So even if the virus continues to evolve, there is this immune backstop on these mutationally constrained regions that continue to provide good immune protection. So just left to make some acknowledgements. I really like to thank the director of the Reagan Institute, Bruce Walker, who's also a former postdoc mentor. I would also very much like to thank Shiv, who's given a lot of wonderful scientific advice for both of these projects and for inviting me to speak at this seminar. To Nusha Nathan and Clarity Kaseke in my lab really led these studies. To Elizabeth Rawson, who I work with to establish the network-based method in HIV and was involved in our COVID studies. To Vivek Narambai, who I work closely with on the Omicron study, and then to all of the patients who participated in the studies, and then of course to the funding bodies as well. So thank you very much for your attention, and happy to answer any questions.
Thank you, Dr. Gaya. Just one quick question from me. Does infection down-modulate class 1 on the cell surface?
I believe it does. I think it can eventually do that through ORF8. So that that infection would certainly have an effect on some evasion from T-cell responses, certainly. But I think what we've seen consistently is that induction of T cells, the recovery of T cells in some cases after initial lymphopenia is definitely associated with survival in more mild disease. I think there's clearly that correlation. But now emerging studies that patients who have pre-existing T cells to some of the epitopes, particularly those in NSP12. So there was a study looking at high-risk healthcare workers, and those individuals seem to more of an abortive infection. So I think that there is a possibility of immune evasion through class 1. But there's definitely, I think that can be either, really circumvented, or if there's a robust T cell response there early on, it will still be able to clear. So it's that constant battle, but I think the T cell response can win out and achieve viral clearance if it's there in a robust manner.
And do we know if it varies across different strains of viruses, or the degree of down modulation, or do we know that?
I don't know that. I don't know if there's been a lot of mutation with ORF8. I'm not aware of that. And so I don't know if across the different, say, variants, if you're seeing different degrees of down modulation. If that's the protein that's mediating, I think you'd have to see some kind of mutation actually having some kind of effect, but I have not seen that yet. But it's a very interesting question.
Okay, thank you. Dr. Kesarilal?
Hello.
Hello.
Please go ahead, Dr. Lal.
Okay, thank you. Thank you very much. It's excellent talk. One thing which I just curious that the example which you gave in HIV as well as the SARS-CoV-2, if you look at the lymphopenia, in both the cases you have lymphopenia and trouble. And also if you look at vaccination profile, there are a good fraction of the patient individuals who did not respond to T-cell response. I mean, something like 21% in your data. Although the amount of number of individual you have was not that great, to look into it in a statistical broader way. So what I wanted to ask you is that the lymphopenia, less T-cell proliferation, which is viral antigen specific, what is the, how these correlate together in terms of looking at how if vaccine is going to be dominating? Do we need to worry about in the individuals where there is no responding T cells, or the correlation with the high severity of the disease with COVID-2?
I just want to make, Dr. Lal, I want to make sure I understand your question. So your question is really just, is it we're trying to relate just the degree of T-cell reactivity and then clinical outcome? Is that kind of your question? I just want to make sure I understand.
Right, so that's one aspect, and also lymphopenia in terms of loss of T-cell in the severe patient. So is something virus has to do with to contribute in the lymphopenia, and so as in vaccination case, there is a non-responding cell. One of the example you said that HLA and its correlation. Is that, or is there something else also contribute in this?
Oh, I see. Okay, so I'll respond to that second question first. So, you know, in the context of these assays in this study, these were individuals who had not been infected at that time. So these are either had recovered disease or had just been vaccinated, or both, or boosted. So at that time, we were just looking at whether the cells that responded to spike, did they have the potential to respond to Omicron in a similar way, or was it different? And that was, so it's possible that during the time of infection, you could imagine there could be some effect on the T cells and you get some lymphopenia, and that would contribute. I think certainly if you lack a T cell response, or in those individuals with a reduction in their reactivity, they would be more at risk for potentially developing more severe disease, or potentially having that lymphopenia manifest, which I see more of a consequence of infection that kind of gets going than necessarily something you would see up front. So I think the reason why we saw the reduction in the Omicron reactivity in these patients who were non-infected at that time, I think is largely driven by, you know, HLA, the epitope specifically binding T-cell reactivity. Although I think you raise a good point that potentially at the time of infection, you would have a reduction in the T-cell reactivity that could be even useful. And I was kind of getting back to one of the initial questions that was asked. I would say in the case of HIV, you know, those individuals have very low Cd4 counts when they get progressive disease. The immune response becomes very dysfunctional. Really what we're trying to do in those cases, either treat them so you have some restoration of the immune response and then come in with a T-cell vaccine that can continue to suppress, or use this alongside antibodies as a prophylactic vaccine. So you get both antibodies, but then also T-cells targeting these constrained regions, so you can really mitigate against variation.
All right, thank you.
Dr. Pillai, you have a question?
Yeah, I do, but I can wait for the others since I can always talk to him. Yeah.
Okay. So where did the question?
I saw some in the chat.
Yeah, there's this one. Let me just read this out. Is it possible that heterogeneity in HLA distribution across geographical regions influences the emergence of T-cell escape variants, specifically in certain regions with susceptible populations?
Yeah, I think it's a fantastic question. I think we're starting to see more literature about actual immune escape from T-cell responses. So I know that there's a current paper by Antonio Bertoletti, which is describing this, where some of these actual spike residues are in fact, really leading to escape from T-cell responses. There's also within Omicron, there seems to be a dominant HLA A29 response that definitely seems that you can get mutations and complete loss. And that's relevant because A29 is one of the immunodominant one of the dominant alleles. About 25% of individuals in, say, sub-Saharan Africa who actually are A29 positive. And they seem to preferentially target this epitope in the ancestral, and then the Omicron mutations lead to profound escape. So I definitely think that there's a potentially component of the impact of HLAs within these populations actually driving T-cell escape that has not been fully accounted for or understood. But I think we need to be thinking about in the same way we think about antibody escape, because it could continue to evolve to become more of an issue.
Thank you. Dr. Mohanty has a question.
Yeah, see, very nice network analysis. See, it is understandable that something which is a hub epitope with a lot of connectivity, it has a, if it mutates, the spike will be destabilized. But but when it binds to HLA, it's still a linear peptide. But where do you find higher affinity for HLA? Did I understand correctly that?
I do, and I think it's two separate processes, right? So I think that so it's two orthogonal attributes of that epitope. So being networked doesn't necessarily mean you're going to bind HLA better. Okay. But we were trying to find those epitopes that were networked, but that also bound HLA. And so we lost the number of good networked candidates in that process because they didn't bind to HLA very well. But we're looking really for those dual properties. And we feel like those would be the best vaccine candidates because they could bind HLA well, be immunogenic, but then also really direct the T-cell response to these networked regions within the viral proteins.
Right. Okay, thank you. Very nice.
I think Rama, can I ask her, allow her to talk? Rama, can you unmute, please? Rama, you can ask a question if you unmute. Yeah, go ahead, Rama. Go ahead.
Hi.
Go ahead.
I'm not sure I'm visible, but okay.
Go ahead.
Hi, this is Rama from Ashoka. Thank you for a very nice talk. Yeah, can you hear me? Am I audible?
Yes, go ahead.
Okay. All right. Thanks, Rahul. Okay, so my question is this. You said that you see memory dropping off, right? You see fewer memory CD8 T cells. But is it likely that there's redistribution? I mean, of course, it's tough to address in humans, but I was wondering if you saw any homing markers early on at the effector stage. Again, this has to be done longitudinally with prior planning. But the idea is this, that if you see some homing markers earlier on during the effector stage, Dave Massapost has a paper in, I think, 2006 or something, the yellow fever vaccine, where you can find hints that at the effector stage that these cells can actually home to other tissues, and maybe that's where they're located. So what you're seeing is a redistribution and not a decrease. Are you thinking along those lines, or?
Rama, I'll just clarify the experiment, and maybe that could help me answer your question, because I think you do raise an important point. But the way we were doing that is we were taking peripheral cells, and then we actually were just incubating those same cells from the same sample at the same time point with either the wild type spike pool or the Omicron spike pool. So the question is less about, when I say drop off in memory, it's really more the memory T cells that will maintain recognition of the spike given the mutations in Omicron that will then expand and proliferate. So at the exact same time point. So the question in that case is more the given T cell receptors, are they able to engage peptide HLA and then expand? Because we're really looking at the exact same point in time. So unless the thought is that somehow the Omicron specific T cells are for some reason redistributing and going to different sites, but because these individuals have never been exposed and either had vaccination or infection with the ancestral strain, I think we have some confidence that really if they were able to maintain reactivity, they would have proliferated to the same manner. Again, we see that in a lot of the patients. There just seems to be a subset in which that similar reactivity drops off. And so that memory T cell response doesn't seem to be as reactive to the Omicron as it is to the wild type at that kind of contemporaneous time point.
Okay, yeah, thanks for clarifying. Actually, I misunderstood your experiment, but yeah, thanks for clarifying. That's great. Just one, I'll sneak in. I'm an ex-NII, product, so I hope they let me sneak in another small question. Your memory cells, do you see any CD45 or any phenotyping done? I'm just specifically asking to see CD45RA and CCR7 expression coming back up.
Great question.
That's my question.
Yeah, so we have not done that. We actually are doing that. We're kind of trying to map the epitopes and further characterize this T cell. So we will have some data coming in that respect. This one, I will say we were a little bit in the race to kind of get the story out. So what I presented was from kind of some very rapid work done in a short period of time, but we will have some more of those details coming for sure.
Thank you so much.
Just before we move on, just one small question. Dr. Gaya, degranulation always correlates with gamma interferon in your experiments?
No, definitely not. So I think that gamma is maybe just good for overall looking to see reactivity and to quantify to some degree. But that's actually why I really like the proliferation assay, which is a 6-day assay, and where you actually will acquire a lot of the perforin and granzyme to be cytolytic. We see a very high correlation between proliferative capacity and subsequent cytotoxicity. So I think as a single metric for function, proliferation is really, I think, the best metric. I think it's better than polyfunctionality in cytokines, definitely better than just interferon or other metrics, and happy to discuss that at a later point. But I think it really is a quite powerful metric for understanding T cell function.
Hi, Rahul. May I ask you questions very quickly? This is
okay. This is the last one. Please go ahead.
Yeah. Hi, Dr. Gaya. This is really excellent talk. Just a very quick questions regarding the peptide MHC of the peptide epitope determinations experiment. There you use a particular antibody. Is it the specific for the whole peptide MHC or just the MHC only?
Yeah, great question, Dr. Singha. So we used just a pan-HLA antibody. So I'm sure you're aware of the W632 clone, which just binds to all HLA alleles. And so what we found is that actually, if you just take the cells and let them incubate at 37, there's just no surface HLA expression. In some of the tapesin-independent alleles, you get a little bit of expression, but just generally it's very low. And then when you put them at 26 overnight and you add in a peptide and you get good stabilization, I think I was able to show with the data, you get really beautiful surface stabilization, which you detect. But because we use that pan-HLA antibody, allowed us to do this in a high throughput manner where we could test any peptide across our entire panel of cell lines and really get kind of robust, reproducible data that way. So that's why we chose it specifically, and it allows for this kind of broad recognition, broad utilization. But it worked well because at baseline, those cells just did not express very much HLA on the surface.
Great, thank you. It's very nice.
Thank you very much.
Okay, thank you so much, Mr. Gaya. We can possibly come back towards the end if there are more questions. It's really interesting talk. Thank you. Okay, we move on to the last speaker, Dr. Tirumala Devi Kaniganti. Dr. Kaniganti received a PhD from the Osmania University in India, then trained at the University of Wisconsin, the Ohio State University, and University of Michigan. She joined St. Jude's Children's Research Hospital in 2007. Dr. Kaniganti is a member of and the vice chair of the immunology department at St. Jude's Children's Research Hospital, and where she is the Rosemary Thomas Endowed Chair. Her lab has made critical contributions in the area of inflammasome biology. She identified the master regulators of inflammasome activation and cell death, pathways of pyroptosis, apoptosis, necroptosis, leading to pioneering advances in the concept of the description of panoptosis and its role in health and disease. She's also made significant contribution in describing the pathological roles of cytokines, that cytokines play in infectious and inflammatory diseases and in cancer. In part, her work involves understanding of the pathology and management of the life-threatening cytokine storms. Her studies, for example, have indicated that TLR2 and MYD88 expression are linked with the production of pro-inflammatory cytokines. Dr. Kaniganti.
Thank you for having me. So I'm going to talk about innate immune responses. inflammatory cell death, the new concept of panaptosis in COVID-19. So first, I would like to thank my lab members who have done extraordinary work all over these years, like for the last 15 years, trying to understand the role of innate immunity and its role in inflammatory cell death. And sometimes it's against the dogma. So you'll see, what I meant by that. So thanks to my previous and current lab members. And then we are constantly exposed to a wide range of pathogens, be it bacteria, viruses, fungi, parasites, including emerging and re-emerging pathogens. So how do we detect this wide range of pathogens? And that's the topic that I'm going to cover. But the main aspect I'm going to discuss is about COVID-19. But to get there, we need to understand, I need to give a brief overview of this innate immune sensing system, the cytosolic node-like receptor family member, NLRP3, and how our studies on NLRP3 inflammasome led to the discovery of panaptosomes and a new cell death pathway called panaptosis. And then it's linked to inflammatory diseases, including COVID-19. So first, how do we recognize this wide range of pathogens? So we have these pattern recognition receptors that are expressed on several of these innate immune cells, such as monocytes, macrophages, NK cells, neutrophils, dendritic cells, and so on. And when you look at the single cell in a close view, so we have toll-like receptors on the cell surface. For example, toll-like receptor 4 that can sense LPS from gram-negative bacteria, toll-like receptor 5 that can sense flagellin again from gram-positive bacteria, and other toll-like receptors that can sense gram-positive cell wall components. So these toll-like receptors sense pathogens on the cell surface or in the endosome. And then there are a large set of innate immune sensing molecule or the family called node-like receptor family. It's a very large family, 22 members in the genome. The other cytosolic sensors include RIGI, MDA5 MAVs, and cGAS-STING. So most of these pattern recognition receptors, like toll-like receptors, RIGI-like receptors, or cGAS, they all sense a unique pattern, that is pathogen-associated molecular pattern or damage-associated molecular pattern. However, this large set of NLRs, we've been studying for the last 20 years, but still we do not know much about, these family members except a few. So when we first started working on these cytosolic, so-called innate immune sensing family, NLRs, they're like NOD1 and NOD2 are the only members that were known to activate NF-kappa B and MAP kinase in parallel to toll-like receptors. So apart from that, not much is known. That's when we started looking at these proteins and asked which one would be a good one to focus on to study in depth. And we picked NLRP3. So why NLRP3? Because mutations in NLRP3 gene causes so many autoinflammatory diseases. So this study published in 2021 showed for the first time that mutations in a new gene, at that time they didn't know the existence of NLR family members or inflammasomes or nothing like that. So they say it's a new gene encoding putative pyrin-like protein, which is nothing but our NLRP3, and mutations in this gene causes several of the autoinflammatory syndromes. So these patients or children with mutations in NLRP3, without any infection, they develop this severe inflammation, like they have joint deformities, rash on their body, like skin inflammation, brain inflammation, eye problems, but nothing is known except that there are mutations in this NLRP3. So that's where we began trying to understand. So we have generated a mouse that is deficient in NLRP3 and screened for multiple pathogens, PAMs and DAMs. And of all the PAMs and DAMs that we screened, we found that bacterial RNA and imiquimod, a small antiviral compound, can activate caspase-1 through cryopyrin or NLRP3. So this is NLRP3, but at that time, the protein is called cryopyrin, a fever-inducing molecule. So here in this study, we showed that NLRP3, the NLR family sensing member, can bind to ASC, the adaptor protein, and then which in turn can bind to caspase 1, the inflammatory caspase. And this complex, once formed, will activate, maturation of pro-inflammatory cytokine IL-1 beta and IL-18 into their mature form, inducing inflammation. So if you think about these cytokines, there's a lot of discussion about cytokines. So I wanted to clarify it. Even small amounts, nanograms, picograms, the smallest amounts of IL-1 beta and IL-18 can induce fever. So these two are fever-inducing cytokines, and the smallest, minutest levels is good enough. You don't need to measure at the nanogram or picogram levels. So what is an inflammasome? At that time, Yerkshop, just a couple of years previous to this discovery, gave a term inflammasome to this platform, triggering activation of inflammatory caspases and for processing IL-1 beta. So this is inflammasome. But then we and others, including Vishwa Dikshit and Yerkshop himself, showed that multiple components like ATP, MSU, and microbial components can activate this complex, driving inflammation via pro-inflammatory cytokines. But we focus always on physiological relevance of anything that we study. So these pathogen-associated molecules like the PAMPs and DAMPs, mostly DAMPs were the ones that were thought to be activating NLRP3. So we wanted to identify a live pathogen, a specificity for live pathogenic infection for NLRP3. So what are the live pathogens that can activate NLRP3? And if there is any physiological relevance for this molecule. So that's when we started looking at several of these bacterial pathogens, viruses, fungi. And from most of the pathogens that we screened for activation of this NLRP3, we found that influenza virus is one that can activate this inflammasome, specifically to induce IL-1 beta and IL-18 as well. So this study established that a live pathogen, influenza virus, can activate NLRP3 inflammasome trigger. And in our quest to further understand this inflammasome, we were able to show that it plays a very important role in infections, including fungal infections, in cancer, metabolic inflammation, joint inflammation, CNS inflammation, autoinflammatory diseases, of course, and so many other diseases and inflammatory conditions. We couldn't believe this, but this is true. Maybe we published 100 manuscripts on just this NLRP3. It's crazy, but this is real, because if you see the companies that are interested in, I think every company, mother, father, sister, are all working on this, and inflammasome activators and inflammasome inhibitors for clinical application. So physiological relevance is huge, and we didn't focus really on finding these compounds because we left that to industry. But in our lab, we were really interested in understanding the regulatory mechanisms. So how NLRP3 can be activated. Why in the world there are so many PAMs and DAMs that can activate NLRP3? It should be a receptor. So is it a receptor? How can this be a receptor if it can sense wide variety of these PAMs and DAMs? So there were so many questions. We didn't believe that NLRP3 is actually a sensor, a receptor, of course sensor, but not a receptor for a specific PAM or DAM. So this leads to the second part of my story, where studying NLRP3 inflammasome led to the discovery of a new multiprotein complexes called panoptosomes. So we started asking, before we go there, I just need to give a brief overview of program cell death pathways. They're very complicated, but I'll try to simplify. There are three major program cell death pathways. So one, of course, is the one that's mediated by inflammasomes activation. So upon inflammasome activation, caspase 1 via gasdermin D induces pyroptosis. So pyroptosis is gasdermin D caspase 1 dependent cell death. And then our apoptosis. I think no one needs introduction to apoptosis, which is non-inflammatory, and it includes so many caspases activation. And then there is another pathway called necroptosis that is regulated via RIP1, RIP3, and MLKL. So these are three major programmed cell death pathways, and that were all thought to be completely independent. And pyroptosis, it's a signature. Genetic evidence is there for caspase-1 gastromin. Similarly, all these genetic evidences and molecular evidences point to the specific pathways that they are very unique and activated upon a specific trigger. So our studies, in our quest to understand what is the substrate of caspase-1, we ended up identifying caspase-7 and PARP. This was against the dogma at that time, because these studies goes back to 2008 and 10. In our proteomics analysis, we were able to show that caspase-1 downstream of inflammasomes can actually cleave caspase-7 and PARP. We did provide genetic evidence to that, but we couldn't find the physiological relevance. Why these caspases are cleaved, we didn't understand. And because there is no phenotype associated except for this cleavage. But then what we're embarking here is on that there is crosstalk. There is no strict tunnel vision for these unique cell death pathways, because we have seen how inflammatory pyroptotic molecule caspase-1 can cleave molecules in apoptotic pathway. And we also know that caspase-8 inhibition leads to activation of necroptosis. So this much more beyond and above going on with cell death, and our studies were connecting this different cell death pathways, and we're trying to understand the basis, because a lot is I think missed in the field because of redundancies or crosstalk. So in our first study, we were trying to understand the importance of NLRP3 inflammasome. So we developed a mouse model of bone destruction, and we were able to rescue that bone destruction by blocking IL-1 beta. So IL-1 beta deficiency in this particular mouse led to complete protection, and we thought caspase-1 and NLRP3 could be involved, but our data was totally negative, suggesting that caspase-1 and NLRP3 are not. And in our search, we did generate like 40 different knockout strains, single, double, quadruplicate knockouts, to just understand the basis of this disease and IL-1 beta maturation. And to our surprise, in a very strange and against the dogma, we found that caspase-8, apoptotic caspase, is playing a redundant role with caspase-1, which is inflammasome caspase. And when we deleted both caspase-1 and caspase-8, or NLRP3 and caspase-8, then we were able to protect fully the mouse from osteomyelitic disease. Of course, the title of this manuscript is very different, because we were able to identify also how diet and microbiome affect these caspases and disease progression. And in another study, trying to understand what is upstream of NLRP3, we also identified that FAD and caspase-8 mediate both priming and activation of NLRP3. Again, this is so much against the dogma at that time, got reviewed in several major journals, but ended up in JI. But still, you will see after 10 years that everybody now agrees that caspase-8 is involved in inflammasome as well. So anyway, overall, what we have been seeing is that there is crosstalk, plasticity, and redundancy between these cell death pathways, and putting caspase-8 into inflammasome pathway. So this is what we have done. I mean, our studies suggested that this pyroptotic pathway can actually activate apoptotic caspases, and then apoptotic caspase, which is caspase-8, can be upstream of inflammasome, upstream of caspase-1. And then there are also in other settings redundancies between caspase-1 and caspase-8. So pyroptosis, apoptosis, necroptosis, 3 independent pathways, but we're seeing intense crosstalk and cross-regulation and redundancies. So we thought there must be a unique other cell death pathway that could be like, using components of this machinery of cell death, be it pyroptosis, apoptosis, necroptosis, but the components of P, A, and PAN might be there. So is there such a complex, or we are just imagining things here? So here is, the definition again to remind us. Pyroptosis means inflammatory caspase is dependent, causes maturation of cytokines, IL-1 beta, IL-8. Necroptosis means RIP1, RIP3, MLKL dependent. And apoptosis is totally a non-inflammatory programmed death pathway. So is there any complex that uses all of these different components? So we were imagining this complex where the components of apoptotic pathway, components of pyroptotic pathway, apoptotic pathway, and necroptotic pathway via their protein-protein interactions, because they have CAR domains, pyrin domains, dead DD domains, rim proteins. They all can form one single complex upon a particular sensor engagement, upon a particular trigger. So we were thinking that there might be a trigger that is activating a sensor. That a sensor might be assembling this complex. This complex molecules could be from pyroptotic pathway, apoptotic pathway, or necroptotic pathway. And we wanted to use not a DAMP or a PAMP, we wanted to use a live pathogen, which is influenza virus for physiological relevance. And by then we have done extensive studies to understand whether influenza virus can actually activate cell death. And by electron microscopically, we were able to see different signatures that it could be inflammatory cell death as well. So IAV induces cell death. These are wild type macrophages. So to test if it is pyroptotic pathway, the way we do is we take NLRP3 deficient mice, caspase-1 deficient mice, and gasdermin D deficient macrophages, and ask if they're required. So if they're required, cells that lack these pyroptotic components should be totally resistant to cell death. However, here we see that wild type, of course, there is cell death. However, even in the absence of pyroptotic components, there is still cell death similar to that of wild type. So this is so strange because we have seen inflammatory cell death by our electron microscopy. So we thought the cell death that's induced by IAV is not pyroptosis, but could be necroptosis. So to study necroptosis, we use RIPK1 deficient cells or MLKL, the executioner of necroptosis. Even in the executioner of necroptosis and RIPK1, in the absence, there is still cell death ongoing. So we are seeing inflammatory cell death, but the inflammatory cell death pathways, pyroptosis and necroptosis, are not required. So what is going on here? This was driving us totally crazy. So it's a long story, and we were able to identify, this is not, identify that. Before we know, it's a long story, as I said, we screened close to 200 different knockout strains, including different TLRs, NLRs, adapters, TNF receptor pathway, interferon, RIGI, MDA5 MAV, CGAS, caspase strips, 200 different knockout macrophages to screen for cell death induced by influenza virus. So a cell death molecule that's involved, if we delete that molecule, it should be resistant. So to make a really long story short, of all the molecules that we screened, when we don't have this ZBP1, so-called ZBP1, in the absence of ZBP1 in macrophages or fibroblasts, there is absolutely no cell death. We can increase the dose, whatever dose you want, and virus is replicating crazily because we can check by Western blotting the viral protein. There is virus replication, but there's absolutely no cell death in the absence of ZBP1. So, and actually we started looking at this because we know NLRP3 inflammasome gets activated by IAV. So we next asked if NLRP3 is downstream of ZBP1. And indeed, when we don't have NLRP3, there is no caspase-1, no IL-1 beta, no IL-18. And also in the absence of ZBP1, there is absolutely no caspase-1 activation, phenocopying what we have seen in NLRP3, putting this molecule upstream of NLRP3. So what we have here is influenza virus that can induce this cell death. Of course, NLRP3 inflammasome was not required for this cell death, but then, however, ZBP1 was required for caspase activation, downstream of NLRP3. So now the big questions: how does ZBP1 deficiency providing protection from this IAV-induced cell death? How does ZBP1 regulate this inflammasome during IAV infection? And what are the upstream regulators here? Is it direct interaction? Is it indirect interaction? What is the complex? What is the molecule that's involved here? So again, we have done several molecular, genetic, biochemical, using all those different approaches. Long story short, we were able to see that ZBP1 via rim proteins binds to RIPK1, RIPK3, necroptotic molecules, and forms a complex with caspase 8 apoptotic molecule. So once this complex is formed, it is simultaneously activating pyroptosis, apoptosis, necroptosis. That means, in other words, there is redundancy downstream because pyroptotic molecules, NLRP3 was not required, 3,7 were not required, MLKL was not required. However, once this node is activated, then there are redundancies. So the redundancies were identified because when we use MLKL knockout, that is deficient in necroptosis, together with ZVAD-FMK that blocks all caspases, there is absolutely no cell death. And we also generated quadruplicate knockout that mimics lack of pyroptosis, apoptosis, necroptosis, the molecules, the core components that are deficient in all three pathways, and that is also totally protected. So to summarize this part, we were able to identify ZBP1 as an innate immune sensor of influenza virus, and it can also activate in NLRP3. And more importantly, we discovered that ZBP1 is a master switch that can drive simultaneously pyroptosis, apoptosis, necroptosis. So we called it as PA and PAN. It also means all, all three kinds of death, and then induces inflammation. So overall, it took us, I think, close to a decade. We started with NLRP3, and then we discovered influenza as an activating pathogen. And then we showed in vivo relevance. And in our quest to understand what is upstream of NLRP3, we discovered ZBP1 and this new cell death pathway that's engaged by ZBP1. So we started with asking, is there a trigger? Is there a sensor? Is this an imaginary complex? So indeed, it's not an imaginary complex. It's formed together with NLRP3, caspase 8 TRP3, engaged by ZBP1. And they all form a spec called panoptosome spec. And several, you might ask questions like, is it real? Do they come together in an IP? So indeed, they all interact. That means especially a RIP3 molecule, which is important in necroptosis, can interact with apoptotic molecule caspase 8, which in turn can interact with pyroptotic molecule ASC. So these three molecules. And then they form a spec. Like you can see beautiful colocalization of caspase 8, RIP3, ASC, pyroptotic, apoptotic, necroptotic components. So, and then our most in recent studies, we were able to discover also another panoptosome, which is AIM2, that is assembled upon HSV and Francisella bacteria. So overall, we have shown that IAV induces ZBP1 mediated panoptosis, Francisella HSV AIM2 mediated panoptosis. We have shown how TACI inhibition can lead to RIP1 mediated. And then in addition to IAV, we were able to discover also that KPT, nuclear transport inhibitor, together with interferon, can activate the ZBP1 pathway. And we also discovered the negative regulators. So all in all, what we have is this platform or a complex that is caspases containing complex with or without inflammasomes or rim proteins. And this platform allows for plasticity in cell death pathways that interconnects pyroptosis, apoptosis, necroptosis. And then we identified the fundamental redundancies observed in panoptosis that cannot be accounted by for pyroptosis, apoptosis, necroptosis. In other words, we cannot call this cell death simply pyroptosis, apoptosis, or necroptosis because they're very unique, engaged by unique pathogens. So now the COVID crisis. And it's very urgent, I think, to really understand the pathogenesis of coronavirus. So why is SARS-CoV-2, the initial strains, were so deadly that induces multi-organ failure? And then this fundamental understanding is important so that we can also address not only COVID-19, but future pandemics. Is there any signature? Is there any innate immune cell death signature that can predict this inflammation and future pandemics? And what is the role of this innate immune system? What are the sensors involved? What are the signaling pathways involved? And so many different questions. So this is the part. So again, to recap, what we know about SARS-CoV-2 is upon lung infection, in those initial studies were all about cytokine storm. You just open any paper, it's all about cytokine storm. So cytokine storm, that's leading to systemic inflammation, morbidity, and mortality. But apart from the SARS-CoV-2, cytokine storm, morbidity, mortality, the remaining is all black box. But we know that there must be innate immunity involved in sensing SARS-CoV-2. And innate immunity triggers cytokines, correct? Like inflammasomes trigger IL-1 beta, IL-18. and then TLRs trigger TNF and other cytokines. And what we were hypothesizing is that the cytokines might somehow induce this inflammatory cell death. Because for this cytokine storm, for this morbidity and mortality to happen, it's not going to happen like that. There must be some inflammatory cell death that's ongoing that might be the cause. So understanding the mechanism of cytokine storm was extremely important to us so that we could identify therapeutic targets. So fundamental questions to investigate. What are the pro-inflammatory cytokines produced in response to SARS-CoV-2? What are the specific functions of these cytokines? And do these cytokines trigger inflammatory cell death? So we looked upon the top, cytokines. Again, as I mentioned, it doesn't mean the amounts, even small amounts can induce whatever the functions that they have assigned for. So we looked at these top 10 cytokines, not only because they're induced highly in COVID patients, but also based on our hypothesis, we were thinking that there must be cytokine combinations that's inducing cell death. So you can see here, we made a cocktail of 10 or 20 different cytokines. And as soon as we added this cocktail, cells started dying. You can see every single cell was dead by this cocktail. And then we separated this cocktail to identify what are the cytokines that are killing these mouse cells or human cells. And we were able to see that they're none other than interferon gamma and TNF. However, TNF alone in small amounts, did not kill cells, and interferon as well. But the combination was so lethal, it induced severe cell death. You can see cell death was up to the roof. However, TNF alone, the amount of TNF we used didn't kill cells. Interferon didn't kill cells. And this, whatever we did in mouse is, mimicked by human cells as well. And please remember, innate immunity is highly conserved, and the genes are all highly conserved. So what do we know about TNF? It can be induced upon TLR engagement or in cytosolic sensors engagement. It's activated by NF-kappa B. It's also a pyrogen, induces cachexia, inflammation. And interferon gamma, you all know that it's a type 2 interferon, critical for both innate and adaptive immunity against viruses. And aberrant TNF and gamma expression has been associated with a number of autoinflammatory and autoimmune diseases. So we wanted to ask, does this TNF and interferon gamma mediated killing that we see in vitro, can it be mimicked in vivo? Can they mimic COVID symptoms? So we were thinking, if this TNF interferon gamma is inducing cell death, leading to cytokine storm, and leading to this death toll, if we inject recombinant cytokines, the mice should be dead like people. So indeed, within 8 hours upon TNF and gamma injections, every single mouse was dead. However, TNF alone or interferon gamma alone did not kill any mice. So next we wanted to ask, what is this nature of cell death that's induced by these cytokines, TNF and interferon gamma? And just to recap, different cell death pathways, they're all tunnel visioned, dogmatic. We cannot think about a common pathway. We have to study step by step, one cell death at a time. So we asked, is it pyroptosis? Is it apoptosis? Is it necroptosis? How do we study that? By using genetics and biochemical approaches. So TNF together with interferon gamma activated pyroptotic pathway. Activated gastromin E, and then also activated, you can see beautiful activation of caspase 3, caspase 7, caspase 6, caspase 8, all caspases are activated. And then also necroprotic pathway molecules, MLKL phosphorylation is there, RIPK1 phosphorylation is there, suggesting that all 3 pathway molecules are activated. But then we asked, as I told you, we generated panoptosis knockout. Panoptosis knockout means a knockout that is deficient in key components of pyroptosis, apoptosis, necroptosis, the quadruplicate knockout that is deficient in caspase 1, RIPK3, and caspase 8. So upon addition of TNF and interferon gamma, cells were dead like nothing. And they were totally dead. However, in the absence of these key components of panoptosis or panoptosis knockout, there's absolutely no cell death. So this is the whole story. I don't have time to go through. What we have identified is that TNF and gamma can activate STAT1. STAT1, which in turn activates IRF1 specifically. There are so many IRFs, but not any other IRF. Only IRF1 via nitric oxide induces this panoptosome formation. and leads to panaptosis, which is inflammatory cell death. Now, if this is true, if we inhibit TNF and gamma, there should be protection from mortality, correct? So that's what we asked. So first step, TNF and gamma injections induces death. And when we used antibodies that can block TNF and gamma, there was 100% protection. Of course, it's blocking TNF and gamma, right? So we asked the other model, this HLH model, hemophagocytic lymphohistocytosis model. Poly-IC plus LPS mimics that model. And when we used antibodies against TNF and gamma, there was 100% protection from this mortality. So we next asked, is it protective in SARS-CoV-2 infection? We use the highest dose of SARS-CoV-2, of course, wild type mice, like were dead, but then by blocking TNF and interferon gamma, we're able to see 50% protection. And not only all these different inflammatory diseases, also in septic shock, in LPS shock, we were able to see 94% protection, from death. So all of this suggests that TNF and gamma induces cell death. And if we can block these two cytokines, there is protection from cell death. So again, I'll just summarize this data. So SARS-CoV-2 infection leads to activation of innate immunity. What pathways that we are still working on. We did publish a few studies, but I'm not going to talk about those receptors. But what we know is that innate immunity triggers TNF alpha and interferon gamma production. As soon as we have smallest, minutest amounts of these two cytokines, they're killer cytokines. They induce this inflammatory cell death called panaptosis. And this induces this storm, which leads to multi-organ systemic failure, and then cytokine shock syndromes might also could be because of these two. And then overall, we're able to show that by neutralizing antibodies to TNF and interferon gamma, we can provide protection. And we also showed that JAK inhibitor, baricitinib could help. And actually, indeed, afterwards, there are clinical trials for baricitinib, and they were able to show that baricitinib provides protection from SARS-CoV-2. And we can also use nitric oxide inhibitor, RIPK1 inhibitor, and the molecules in panoptosome and panoptosis inhibitor. So from this study, we learned so much about inflammatory cell death that's driving the cytokine storms and pathway molecules that could be targeted. So in this review, I think we all use cytokine storm as an umbrella term. No one really knew the definition of cytokine storms. So we had an opportunity to define cytokine storm, and we defined it by suggesting that this innate immune mediated inflammatory cell death drives production of cytokines, and the cytokines can lead to these cytokine storms, right? So cytokines TNF and gamma induces inflammatory cell death panaptosis, which can lead to cytokine storms. And this will be applicable for so many of cytokine shock syndromes, be it with viral, SARS, MERS, IAV, SARS-CoV-2, and bacterial sepsis-inducing molecules. And even in therapy, CAR T-cell therapy or autoinflammatory diseases, that could be very well applicable. And in disease settings, so we were able to show that upon infection, viral and bacterial infections, distinct panoptosomes are assembled. So with IAV, it is ZBP1 panoptosome, and with Francisella, it is AIM2 panoptosome. And then we also have shown cytokine storm by these particular cytokines can induce panoptosome. And we published a few other recent studies suggesting its involvement in cancer, because hallmarks of cancer is resisting cell death. But with panoptosis, we can kill those cells that resist death. And in autoinflammatory bone disease, we're able to show the importance of panoptosome. And then many more things are coming up. So what is panoptosome? It's an innate immune cell death complex that can sense danger, which could be pathogens or altered cellular homeostasis, and activates inflammatory cell death, which is panoptosis. So with this, I would like to thank you again for the opportunity to share our work, and I would be happy to take any questions.
Thank you so much, Dr. Kanaganti. And my colleague Somen has some questions.
Sure.
Yeah, so I think it was, first of all, thank you. It was really very enjoyable talk, wonderful talk, I think, Dr. Kanagati. I think I have a quick question about this TNF and interferon gamma synergism, and related to, of course, NF-kappa-B. Normally, we do not see TNF would kill a cell if it is allowed to activate NF-kappa-B pathway. So, and in your study, it clearly demonstrates that TNF interferon gamma together caused pervasive cell death. Is it that have you looked into a possible NF-kappa-B connection? Is it that interferon gamma is somehow reducing NF-kappa-B activation if acting along with TNF, and thereby probably favoring cell death component of the TNF pathway?
I think there are two different entities here. So TNF alone in high concentrations was shown to engage cell death. So we used really minute amounts of TNF that by itself cannot kill cells. So high concentrations of TNF is known to kill cells. So we use this lowest amounts of TNF. So TNF might be produced, via TLRs, because TLRs engaged, they're on the cell surface. As soon as they see any pathogen, they induce NF-kappa-B, and via NF-kappa-B, TNF is induced. And then there are NK cells. I think in our study, we were able to see that truckloads of interferon gamma is produced by NK cells. So if these two cytokines are in the system, that's when it's detrimental. Right. So this is what we think. They're two separate entities, and the combination is lethal. But if they're in the bloodstream, that's where the danger is, right? So that's where I think vaccine research should also look into this panoptosis, because a low level cell death is always good. It's protective. But then bloodstream, like in bloodstream, having these cytokines and cell death is detrimental. So that's the difference, I think.
Okay, thank you very much. Yep. Rahul?
Dipyaman, you want to ask a question? Dipyaman?
Yeah, hi.
Yeah, go ahead.
That was a very illuminating talk, Dr. Kanayanti. So I had just two questions. One is, in the human context, how do you think the stochasticity that we see among the patients in terms of whether they will succumb to cytokine storm or not can be explained? Because I would imagine that the cell death pathways will be activated in all viral infected epithelial cells, right? So whichever that's what your hypothesis is, that the Cd2 expressing epithelial cells which are getting infected, they are undergoing this cell death and then producing the triggering cytokine, starting a cascade. Now, how do you explain the stochasticity of succumbing to this phenomenon? So that's the question. I don't know.
Sorry, your voice was breaking a lot. So can
Dipyaman, even here, can you either type it in or get a little closer to the microphone? Sorry.
Sorry,
I
couldn't hear. Sorry.
This is much better. Go ahead.
Okay. So I was, I think, so your hypothesis is the cell death pathway will affect the C2 expressing epithelial cells which are getting infected, and they are starting the initiating cytokines, leading to the cascade, and that's it, right? Now, how do you try to explain this in the human context, where there is a large stochasticity of which human beings will be succumbing to the cytokine storm phenomenon? So are there elements of stochasticity in this, in induction of this cell death pathway?
Yeah, so as we discussed, I think, not all pathogens can induce the cytokine storm. And there is some specificity, even like for SARS-CoV-2, I mean, the initial strain were inducing this, as you know, severe morbidity and mortality. The most recent Omicron strain is not inducing. So it all depends on the pathogen and whether the pathogen can actually engage this panoptosis and the cell types that are involved. So we know that for most part, innate immune cells like monocytes, macrophages, dendritic cells are involved. So epithelial cells might not really, play a role, but lung epithelial cells do also express this. So we need to study in depth regarding which cells are actually expressing these particular sensors and and like the sensing molecule, we still do not know the sensing molecules that are inducing this particular combination of cytokines, or what are the sensors that are involved. So to keep it short, I think it depends on the strain, it depends on the amounts of infection as well, the dose, the exposure, and of course the underlying conditions. Correct. So it's very interesting that patients, Crohn's and Crohn's disease patients who are on Remicade, which is blocking TNF, they fared well. There are so many studies because the joint combination is bad. But even if you block one cytokine, the other one cannot do this killing job. So I think that's one of the reason, and also baricitinib clinical trials are providing another line of support that if it is detrimental, then these are the pathways that are activated. If it is not detrimental, then we can recover fast, right? So it's not that bad. Maybe panaptosis is not induced to that extreme levels.
Thank you so much. Thanks.
It's a relatively new area. We are learning also.
Gopalan, go ahead. Gopalan.
Yep. Thank you. Can you hear me?
Yes, yes.
Oh, great. Thank you. It's a very nice presentation. Probably a lot for me to digest. Just wondering.
Yeah, it's a lot. Sorry. Yeah.
Yeah. Poor TNF-alpha is so innocent, but as a complex, is anything known biochemically? Do they interact and associate? And could one quantitatively do relative titration to find out minimal ratio stoichiometry that would be required?
Yeah, it could be done. We just stumbled upon, understanding what are the sensors. So we didn't really titrate and, went towards that path. Yeah. Yeah.
I was wondering if molecules interact all the time, do TNF, and if they were to form a complex, maybe they make a different entity altogether.
Yeah, I think they don't form a complex. They just bind to the receptors, their respective receptors, and then engage this
that is a standalone independent mechanism. But I'm just wondering, mixing these two cytokines in a tube in a biochemical sense, if they associate, then they become a third beast altogether. Ain't it?
Yeah, we didn't think about it. Yeah.
Okay. Thank you very much. Interesting, and thanks a lot.
Thank you.
Hi, this is Rama from Ashoka University.
Hi.
Am I audible?
Yes, I can hear you. Yes, please go ahead. Yes, I can hear you.
Rama, go ahead.
I'm not audible. I'll come back.
Yes, go ahead.
Yes, okay, all right. I'm glad the organizers invited you and you accepted, because this is very illuminating. I must confess, I'm woefully ignorant about this area. So these are more clarifications. So for me to go back and read up more. Yeah, so my question is this, okay? Do I have it, or my clarification, my request is this. Do I have it right that I'm picturing panoptosis containing the ZBP1 protein? You said you call it a switch. So am I right that it's the switch that engages all the other three pathways?
Yes. That is in response to influenza virus. For SARS-CoV-2, we still do not know what is the sensor.
Okay. All right. So, and now the other question is, these other three pathways, I'm guessing they exist as some sort of pre-existing molecular complexes within a cell. And depending on the signal and the response to that cell's response to that signal, one or the other is engaged. Am I right?
Yeah. So they are in the cytosol. They are not in the complex form. They're there in the cytosol. So it's an amazing network. So for the receptors, they require upregulation, some of the receptors. So upon their upregulation, then they can form a complex, bind, to caspases to form a complex. It's just a network. They're just staying in the cytosol. Depending on the trigger, they just come together, like a puzzle piece, and then form the complex. So that's formed might be very unique to a pathogen.
Okay, great. So just one final clarification. So then, is it very local, like even the pan-apoptosome formation, or is there a critical number of pan-apoptosomes that need to form, or is it like one forms and then there's like some sort of a chain reaction that leads to panapoptosis.
Yeah. So as soon as in a single cell, panapoptosis is activated, then that cell will die in an inflammatory way. That means it releases all its contents, like spills out, like cytokines and like other PAMs, like dams, like, because in a cell, we have to imagine pathogen when it's entering, it causes damage. And also while it's entering, it's releasing its components, and pathogen is trying to survive. Host is trying to kill pathogen itself. When host kills pathogen itself, also the components are there. So inflammatory cell death is so bad, it releases, like, spills out all of these PAMs and dams that leads to this severe, cytokine storm.
Okay.
And
so
engages
no,
other cells. Yeah, now are getting
I
multiple signals, to die.
I see. Okay, I'll hold my horses. I'll finish that. Thank you so much.
Okay, thanks, Rama. A couple of more quick questions. One from Keshav's Saini. Is there a particular innate cell type that might be primarily contributing to high levels of TNF alpha and gamma interferon?
Yeah, so I think, yes, there might be a particular sensor for cytosolic sensing and inducing this cell death. But for TNF, engagement of any TLRs can do the business, right? So, but TNF alone is not sufficient unless it's produced in truckloads. Okay, so TNF to be in a cell, it's easy. TLR engagement can lead, but the cytosolic, we need another sensor to activate this pathway.
Yeah, but is there an innate cell type you think might be important?
Yeah, so most of these innate cells, like any innate cell you think of, they all express these molecules, and also epithelial cells expresses these molecules. So it's very understudied area. Like, which cell types, how much they're contributing, and what is the dose. Like, it's a beautiful area, but understudied. Always people were thinking innate immunity is a different field and cell death is a different field. But now what we're saying is it's innate immune cell death. That's one entity.
I see. Okay. And the second question is, have you detected direct interaction of IRF1 with ZBP to activate panapoptosis? Or will NO play a role?
No, I think it's a transcriptional, it upregulates molecules involved in this pathway. So it's a transcriptional regulator.
I see. Okay. Okay, I think we're done. I'd just like to briefly thank all four speakers today. It's been a wonderful exposition, both yesterday and today. Very, very insightful and very exciting and interesting.